for Excellence in Regulatory Science and Innovation. Welcome. My name is Joe Ross. I'm a general internist and health services researcher at Yale and one of the co-directors of the Yale-Mayo Clinic, CERSI. And for the past several years, actually since our inception, we've had what we call a CERSI Scholar program. And we gleefully stole this idea from the Johns Hopkins CERSI when we learned about it, which is really just an opportunity for students, fellows, and maybe even junior faculty to highlight the regulatory science research that's being done on our campuses, to provide small stipends that can be used to support the projects, or more often than not, support publication fees in journals when they write them up. And we've always enjoyed creating this seminar that brings together our scholars in the past to present their work and to learn from one another. And a couple of years ago, we opened it up even more broadly to the other CERSIs that are funded through the FDA's Office of Regulatory Science and Innovation, and then having them nominate the scholars that are working under their supervision to present their work as well, to broaden the community and help people learn and meet and network with one another. And we are delighted that there's even a new CERSI coming, the Triangle CERSI, representing the institutions of Duke and UNC, and we're very, very happy to also have the folks from the Harvard MIT Center of Excellence that also does regulatory science research on their campuses. So on behalf of Molly Jeffery, my colleague at the Mayo Clinic, welcome. We have a pretty fabulous day scheduled with numerous presentations from scholars. Thank you in advance to my colleague at Yale, Jason Schwartz, School of Public Health, who's going to be helping to coordinate the day with us. And I'm really delighted to be able to introduce as a keynote speaker Rear Admiral Richardae Araojo, who serves as the Associate Commissioner for Minority Health and Director of the Office of Minority Health and Health Equity at the FDA. I've had the great opportunity to work with Rear Admiral Araojo over the years, and some work that she's done at Yale and some work that we were able to do together as part of the CERSI. Rear Admiral Araojo provides leadership, oversight, and direction on minority health and health disparity matters for the agency. She previously served as director of the Office of Medical Policy Initiatives in FDA's Center for Drug Evaluation and Research, or CDER, where she led a variety of broad-based medical and clinical policy initiatives to improve the science and efficiency of clinical trials and enhance professional and patient labeling. Rear Admiral Araojo joined the FDA in 2003. She received her Doctor of Pharmacy degree from Virginia Commonwealth University, and then completed a pharmacy practice residency at the University of Maryland and earned a master's degree in pharmacy regulation and policy from the University of Florida. Dr. Araojo is going to keynote our scholar seminar series today with some opening comments, and will be available to take some questions before we move on to the CERSI Scholar presentation. So Rear Admiral Araojo, thank you for making the time to join us today, and we look forward to your talk.
Thank you so much, Joe, and it's wonderful to see you. And good afternoon, everyone. I really appreciate the opportunity to speak with you today. And I am going to share my screen, so just give me 1 second so I can pull up my slides. And then I will make sure that everyone can see everything. All right. And hopefully you all are able to see my slides.
Yep, looks good. Thank you.
Great. Joe, of course, really appreciate the opportunity to be here and speak with all of you today. And good afternoon, everyone. I am really honored to join you to help kick off the research seminar this afternoon, and really exciting to hear about the urgent topics stemming from new directions in regulatory science that the 2023 CERSI Scholars are working on. And of course, want to extend my congratulations to all of the CERSI Scholars on the important work that they're doing in answering some of the pressing questions that we face at FDA to continue to make our country healthier. So I'm so pleased to be able to highlight the work that we're doing within the Office of Minority Health and Health Equity, and the importance, of course, of incorporating diversity, equity, and inclusion in regulatory science research. So I wanted to begin today with just providing some information about the Office of Minority Health and Health Equity and the work that we do, because oftentimes, even though we are very much engaged with the CERSIs and work across all of the CERSIs, oftentimes when we are beginning our engagements, many offices and many groups are not aware of the work that we do. So for some background, the Office of Minority Health was actually first established at FDA in 2010. It was the first office dedicated to providing leadership and policy direction for the agency on issues related to racial and ethnic minority populations. And a few years ago, our office was actually renamed to the Office of Minority Health and Health Equity. And we have a dedicated mission where we are focused on advancing, protecting, and promoting the health of diverse populations through research and outreach and communication that works towards addressing health disparities and, of course, ultimately advancing health equity. So our office is within the Office of the Commissioner, which means that we really engage broadly across our agency. And we also work very closely with both public and private sector stakeholders. And as you heard just from our mission, we really focus our efforts in two key areas. So we have our outreach and communication program. That program really works to improve FDA's communications with the populations that we serve. And we do that through a variety of culturally and linguistically tailored programs, initiatives, and campaigns. And one of our goals is really to work to make sure that we have diverse means and mechanisms to reach diverse consumers. Some of the ways in which we do that is through health education materials that are provided in multiple languages. We have broad social media outreach, we have newsletters, we have a website with all of our tools and resources. But we also want to have forums where we can bring experts in minority health and health equity to share information with our agency as well as with the public, including results from CERSI projects and many other types of activities that we engage in. And we've been able to do that through our Health Equity Forum podcast, as well as our Health Equity lecture series. And of course, being that we work within the space of health disparities, collaborations and partnerships are really critical to help us achieve our mission. And of course, we also have our research and collaboration program. And this is definitely an area that's very important when we think about our engagement with the CERSIs. So our research and collaboration program really aims to advance minority health and health equity focused research. And we do this through supporting both intramural research as well as extramural research. And we participate in research opportunities across our agency, of course, being able to engage with the CERSIs as well. Another area for us that is a component of our research and collaboration program is our internships and fellowships. And this is really important because it's our opportunity to be able to not only continue to train those that are working in the space of health disparities, but also to be able to support advancing diversity across the scientific workforce. And again, across all of the work we do within our office, we have broad stakeholder engagement to help advance our efforts. So I want to spend some time talking a little bit about CERSI, of course. This has been really, our office has been engaged with CERSIs for so many years, and it's really been critical to us being able to answer pressing questions. We've been able to engage across all of the CERSIs, as Joe mentioned earlier, and I have only 3 brief projects highlighted here. And it's just an opportunity for us to be able to see the wide range and scope of the type of work that we're able to do and engage with the CERSIs, which has been really important, not only to our office, but across our agency. And through our CERSI projects that the Office of Minority Health and Health Equity has had the opportunity to collaborate on, of course, we're working with across all of the CERSIs as well as with many of the FDA centers and offices as well. You see here just a very few projects. We've engaged in many other projects aside from these, but it highlights that scope. So for example, we participated in a project related to usability testing of virtual reality for pain management. Another project that we have ongoing right now is integrating patient and consumer generated discursive data to inform and enhance FDA One Health initiative communication strategies. And then another project that we engaged on was focused on characterizing population-specific clinical asthma profiles. So CERSI has been really important to our office to be able to really engage on such broad topics, and again, to help answer pressing questions and help answer pressing regulatory science research questions. So of course, through all of our CERSI projects, one key area is to be able to publish the results of the findings, to actually get that data out there. That is definitely a priority for us within the Office of Minority Health and Health Equity. We want to be able to increase data available on the populations that we serve. And one way to do that is, of course, publishing the findings from our work, and also being able to promote that work throughout the different forums that we have. We often bring our CERSI investigators to be able to come and highlight the research that we've collaborated on through the various mechanisms that we have across FDA, including within the Office of Minority Health and Health Equity, like our podcast or our Health Equity lecture series. And what you see here is just a paper that we published with UCSF, and this was on racial and ethnic differences and eligibility for asthma biologics among pediatric populations. So I want to take some time now to highlight some initiatives that we have ongoing across the Office of Minority Health and Health Equity. And hopefully this will give you an idea of the type of work that we do across the office. This is by far not all the work that we have ongoing right now, but these are some key initiatives that are really important that we are working on. And one of those that I'm going to highlight is one that is probably our most long-term notable initiative, and that's our Diversity in Clinical Trials initiative. As many of you are probably aware, the FDA has long worked to advance diversity in clinical trials, and there's a lot of work happening across our agency, across their centers and offices. And we developed our Diversity in Clinical Trials initiative to be able to support FDA's broader efforts. And within this initiative, we have an ongoing multimedia public education and outreach campaign that really works to raise awareness around the importance of diverse participation in clinical trials. The campaign aims to combat myths, to educate consumers about key issues, provide positive messaging reflecting diverse spokespersons who are representative of diverse communities. We hope to be able to stimulate dialogue among peers and peer-to-provider groups, while tailoring our resources to be culturally and linguistically appropriate, as well as provided in multiple languages. Within our campaign, you see a couple of areas here, the different types of resources that we have available. We have public service announcements, we've hosted public meetings, we have videos, podcasts, we have both print and digital health education materials. We engage in social media outreach, and we have a dedicated website that houses all of our tools and resources. I will also highlight that across all of the work that we do across our campaigns, broad stakeholder engagement is very important. I know Joe mentioned earlier ways that our office has engaged with Yale. This is one of those areas in which we've engaged and work closely with Yale, especially with our community engagement activities related to clinical trial diversity. Within this campaign, we've hosted listening sessions, we have ongoing collaborations and partnerships, and we also have ongoing research efforts to help answer some of the questions that we have, and we are constantly working to add new tools and resources. So what you see here is just a snapshot of some of those resources that I mentioned. Of course, we have hosted conversations on our podcast related to clinical trials and clinical trial diversity. We also, as you see here in the middle of the screen, have some components that are focused on specific diseases and conditions. And this is a picture from our Let's Take Charge campaign. This is a campaign that we developed in collaboration with the HHS Office of Minority Health, and it's focused on advancing diversity across lupus clinical trials. And this was a campaign that was also launched in both English and Spanish. And then, of course, social media. It's very important for us to be able to put out through social media a trusted resource where we can put out different key themes and messages and link back to where you can find additional information and where you can learn more. And some of the resources that we link back to are seen here. So we have, for example, our clinical trial diversity fact sheet, we have a brochure, we have our infographic, and we also have a dedicated website that houses all of the tools and resources that we have available. I've talked a little bit about our education, our patient education materials and information, but we also focus on the role of healthcare providers. And we have published on engaging community providers and clinical research. And this was a publication with Dr. Woodcock, as well as Dr. Gary Puckrin with the National Minority Quality Forum. We, of course, know that many strategies have been developed to increase enrollment of diverse populations. And one strategy that we think is important is also engaging community clinicians and research, and really thinking of ways that we can incorporate these providers into the research infrastructure in a sustainable way. And this is what we discuss in this paper. So now I want to move on to another initiative that we have within our office. And this was a really interesting one. It was an initiative that came from a 2021 congressional mandate for our office to educate the public on potential risks of products containing hydroquinone or mercury. The FDA has issued warning letters to companies for selling over-the-counter skin lightening products containing hydroquinone or mercury that do not meet the requirements to be legally sold as OTC drugs. And these letters followed the agency receiving reports of serious side effects, including skin rashes, facial swelling, discoloration of the skin, among other types of reactions. And FDA also issued a consumer update warning the public about using skin lightening products that contain mercury. We know, of course, that in the United States, these types of products are often marketed to racial and ethnic minority communities for any type of treatments, from skin tone to acne, age spots, freckles, wrinkles. And so one aspect for us that was really important was the work that we did with Reagan-Udall Foundation. Reagan-Udall Foundation conducted formative research for us to help us understand the main drivers of the use of these products among diverse racial and ethnic communities. And what was really critical to that research was being able to engage a wide range of stakeholders, including affected communities, experts, skincare specialists, as well as dermatologists. And data from that formative research was used to develop the campaign that we launched. And of course, with any of our educational efforts, we want to be able to continue to learn to make sure that we're refining the information that we put out there. So we also, after launching the campaign, we also announced 2 funding opportunities so that we could continue to expand education on skin lightening products. And so what you see here are some of the resources that are available as part of our Skin Facts initiative and through the public education campaign. We have educational materials that are all available in multiple languages. We have public service announcements, and really important is the fact that we have a partner toolkit with social media messages for organizations and individuals to share Skin Facts information with their communities or their patient communities. So now I'm going to move on to one of our newer initiatives. This was actually, this has really been a really large initiative for the office, and it was developed in FY21. And it really highlights our continued dedication to enhance equity and highlights research projects and communication resources that enhance equity across 3 key aims. One of those is enhancing equity in clinical trials through our efforts to continue to advance diversity in clinical trials, enhance equitable data efforts by funding research and supporting research that increases data available on the populations that we serve. And probably the most important area for us that's been really critical to the work that we do is enhancing equity of voices. So not only are we working to amplify FDA's communications with diverse groups to ensure stakeholders and consumers are informed about FDA's efforts, but also so that we can continue to understand diverse patient perspectives, preferences, and unmet needs to inform our work. So as I mentioned, with our Enhanced Equity initiative, we've had the opportunity to put out multiple funding announcements that have really led to opportunities for us to answer pressing questions related to equity in clinical trials, to support projects that you see some here. This is just a snapshot. We have over 30 projects that we've been able to fund that looked at ways that we can continue to understand barriers and challenges to enrollment in clinical trials across different diseases and conditions. So for example, COVID-19, hepatitis B, as well as opportunities to continue to understand, again, diverse patient perspectives, preferences, and unmet needs. And what you see here is just an outcome of one of those projects where we worked with the Hepatitis B Foundation. And ultimately, the results from that work were highlighted in this publication that was focused on attitudes towards clinical trial participation among people living with chronic hepatitis B. And all of the research projects that we have funded, including across the CERSIs, can be found on our website. And lastly, our aim focused on equity of voices. Again, this is a critically important area for us. And not only have we funded research, again, that helps us to continue to understand diverse perspectives, but we also want to make sure that we're able to support opportunities to put the patient voice out there. And we developed a video series that features a number of individuals living with HIV and their experience with antiretroviral treatment. You see here just a picture of Vincent where he's sharing his story. We have a number of videos that are on our website. And I think what's been so important through the process and working with different patients and patient communities is really being able to hear their personal experience. And it really, I think, helps to remind all of us about why we do the work that we do. And I know I've mentioned this a few times, the opportunities that we've had through our Enhanced Equity initiative have been really important through a number of funding opportunities that our office has been able to announce. These funding opportunities have helped us to really work with diverse researchers and scientists, as well as a wide range of organizations through our Health Equity Innovation Awards that's gone out annually. We've also had a number of COVID-19 and Health Equity Innovation Awards that were announced. And as I mentioned previously, we had 2 announcements that were related to expanding education on skin lightening products. So lastly, I wanted to take a moment to highlight work that we do to help support that next generation of diverse researchers and investigators. And one of the ways in which we do that is through our Genomic Science and Health Equity Postdoctoral Fellowship. This is a fellowship that is conducted with the Office of Minority Health and Health Equity in collaboration with the National Human Genome Research Institute, or NHGRI, at NIH. And the fellowship is really focused on training scientists in genetic, genomic, and pharmacogenomic approaches to advance minority health and health equity. Our current fellow right now, Dr. Raven Hardy, is actually doing work related to sickle cell disease. She has a mentor within our Center for Biologics Evaluation and Research, or CBER, as well as a mentor at NIH. So it's really a unique opportunity where you can work within 2 federal agencies to conduct research within this space. And we're really excited about our upcoming fellow, which is going to be doing work related to rare diseases and working with the Office of Orphan Drug Products here at FDA. So with that, I thank you so much for the opportunity to speak with you today. And of course, please stay connected with our office. You see all the ways here in which you can do that. I'd also, again, like to congratulate the CERSI 2023 Scholars and the important work that you do. I know it's been so important for our office to be able to work across all of the CERSIs, and the work that we've been able to collaborate on has been critically important. So thank you so much for all that you do. And I look forward to continued engagement. Thank you.
Thank you, Richardae. That was great. It's such an amazing, really interesting portfolio of research. Do you have time for a couple of questions in case? Sure. Great. And so please, put up your hand if you have a question. And maybe I can start. Well, first of all, your office is so helpful to us in that we were not on your list of projects, but the acute pain pathways project that we've been doing, where we've been recruiting a diverse, sample of patients for this opioid-related work, and it's just been great to have your office's involvement in that. I'm curious, how well does your office align with not just the HHS office, but also the NIH office in terms of trying to drive these like clinical trial diversity initiatives kind of at the whole government level? Like those seem to be like the major offices having some responsibility for clinical trial recruitment, especially NIH. And maybe you could say a word about what's going on there as well.
And Joe, I knew you were going to say that. And as I was saying that, I'm like, oh no, I didn't have the Yale-Mayo project on there. It's so important, and that we really appreciated the opportunity to collaborate on. It's really important work and answering, again, important questions for FDA. And yes, so to answer your question, we do engage with the other offices of minority health. We actually have a monthly engagement with the directors across the offices of minority health. In addition to that, we have engagement. So I mentioned about our collaboration with HHS Office of Minority Health related to one diversity in clinical trials initiative. But yes, we absolutely engage with NIH. We engage with HHS OMH. We engage with HRSA and all of the other OMH offices. And I think while each of our agencies may have a focused mission, of course, when we talk about clinical trial diversity, it's a very broad area. We know that we need all stakeholders at the table when we are working to advance diversity in clinical trials. Some of our resources have been promoted and some of the NIMHD work that they have and highlighting the resources we have at FDA related to clinical trial diversity. And then, of course, just the collaborations and engagements we've been able to have across the various OMH offices as well on a broad range of areas, even beyond clinical trial diversity.
That's great. Any questions from the group? I can ask more.
I have a question, Joe.
Oh, great.
Yeah, thanks. Really enjoyed that. So actually, I'm more numerically inclined and less linguistically inclined. So this explains my question and also why I can't remember Dr. Lola's last name. So she spoke here at Maryland last week, and I asked her a question about metrics. So, going forward, right, and trying to do a better job with diversity in clinical trials and monitoring that, and presumably we want to think about metrics for whether we're achieving that. And maybe within a study that's easy to do, to use our eyes to evaluate whether we're being equitable. But if you start pooling across studies, maybe it becomes a lot harder. Are people developing metrics for, achieving diversity across multitudes of clinical trials? I mean, how would the University of Maryland know if we're doing well in this regard? Other than using our eyeballs, I suppose, looking at lots of different studies that are on the go.
Yeah, I think that's a really good question. And I'm sure you're probably talking about Dr. Fashionage, Lola.
Yeah, yeah.
Okay.
Yeah.
So I think, we have resources here. I'm sure Lola highlighted those resources. For example, drug trial snapshots, which highlights demographic data from pivotal clinical trials. And of course, I know there are a lot of stakeholders out there. There are a lot of publications where different academic institutions, even CROs and organizations have published and put information out there based off whether research they've been engaged in, work that they've seen that FDA's done, like through snapshots and things like that, where we start to see where they highlight that demographic data. Oftentimes, I think where we tend to think about, especially I'll say from the Office of Minority Health and Health Equity, when that information comes out there, I think you can see pooling of information, but sometimes the pooling of information doesn't necessarily highlight where there might be specific diseases and conditions where we need to continue to do more. So it gives you an idea. FDA put out the 5-year summary report from drug trial snapshots, which was really helpful. You could see some of that pooling of data, but then that was also broke down by different therapeutic areas. It was broke down by rare diseases. Don't ask me, I can't quote right now what the numbers are, but it gave us a general idea of trends. And I think it's important to know those trends, but I think we also have to dig deeper sometimes to look at that data across, maybe it's a specific trial. What worked well? What were some of the best practices from that? And thinking about could those be best practices that we apply to another trial? But I really think you have to think about the disease, the condition. There's a lot of different areas to think about. So I don't think there's one best metric or number. I think there's a lot of work that we need to continue to do, but I do think some of the resources and information out there is helpful in seeing some of those trends. But sometimes we do have to look a little bit deeper because, a number may look great, but we might see that there's additional work we need to do in certain areas.
Okay, thank you.
Richardae, Jim's question reminds me, we have CERSI project that's been going on this past year, led by Jennifer Miller, an incredible bioethicist here at Yale, that it occurs to me, I wonder if your office is not as involved in, or should be more involved in, with working with the Oncology Center of Excellence, focused on oncology trials, essentially estimating participation to prevalence ratios, using sort of baseline demographic data for clinical. Rebecca's just telling me Lola's on that project. So maybe we can give you an update just to tell you where we're at and make sure that because I think Jim is exactly right. This idea of measuring allows us to monitor, which then allows us to improve things. And so Jennifer Miller's been doing some really great work in that regard. So other questions? Jason, I think we're about to jump in with one.
I do have one. I'm eager to hear. Thanks for all the great work that you do. I'm eager to hear more about your office's work during COVID-19, particularly with respect to therapeutics and vaccines, where we know there are unique causes and sources of hesitancy and safety and confidence in government recommendations around among racial and ethnic minority groups. I've seen some materials on your website that you were involved in your office talking about vaccine safety, talking about the development process. I wonder if you'd say a little bit more about your work on that front and how you'd evaluate how those outreach efforts fared.
I think that's a really good question. So there's a lot of work that we did related to COVID-19. I think one of the efforts related to COVID-19 that was important for us, of course, we worked very closely with CBER and Dr. Peter Marks and colleagues across CBER. That was very important for us. And another component that was important for us was really making sure that we highlighted for all of the stakeholders and groups that we worked with that we were a trusted resource for information, and really working to counter some of the misinformation that was out there. We had weekly emails that we still send out, weekly emails that kind of encompass the FDA COVID-19 efforts, education, information. We worked with a wide range of stakeholders that we engaged with that are focused on minority health so that they have those resources to communicate to their patient communities. So that was one of the areas that we focused on that was a priority. Through some of our COVID-19 efforts, we also worked to put out, as you saw, some of the educational materials across many languages, including native languages. We also had one in American Sign Language, which was the first time for our office and something that we plan to continue. So we were really excited about that. And a lot of engaging with community organizations, really organizations that we're always trying to find new collaborators and those that we can engage with to reach diverse communities. And I think COVID-19 really helped us to be able to do that in some respects because we were able to engage with places of worship. We engaged just a broad scope of groups to be able to make sure that they knew that FDA is a trusted resource for information where they could find information on COVID-19 vaccines, truthful information, highlighting the resources we had in multiple languages. And then in addition to that, we were able to put out through funding that we received our COVID-19 and Health Equity Innovation Awards. So that helped us to also start to answer some questions we had related to COVID-19. And we're really continuing that work. So even the education that we have available, the research that we've been able to fund, as well as the, really cross-communicating and promoting the important work that's happening across our agency.
That's great. Any last questions before we get going with the scholar presentations? Well, Richardae, I can't thank you enough. Thank you for making the time to kick off this meeting. It's so great to see you. I look forward to continuing to work with you in your office. And you're, of course, more than welcome to stay as long as you want and hear some of the talks if you're interested. But I think at this point, I will I'll hand it over to Jason, who will lead the first session. Or
Indeed. Now, I'll just a housekeeping question. I know there was a 12:40 to 12:50 break on the agenda, but I'll look for guidance, Joe, from you or others if we want to just forge right into that first session.
Yeah, let's forge. It's Friday. Probably if we can end 10 minutes early, people will be happy. Right?
That sounds great. So thanks everyone. Again, I'm Jason Schwartz from the School of Public Health at Yale, and get the pleasure to contribute to some of the CERSI projects with Joe and his colleagues here at Yale. So as you've seen on the agenda, we have a full afternoon, right? 4 sessions, I think 17 presentations altogether in blocks of 4 and 5 to hear about all the fantastic work that's been happening from the CERSI Scholars. So I'll be leading the first session over this next 50 minutes or so, and we'll hear from 4 of the CERSI Scholars. Just a couple housekeeping reminders. As you know, folks have been asked to limit their remarks to about 10 minutes, for up to 10 minutes, so that we can stay on this full schedule that we have. After those presentations, we'll do them in order, and then there'll be hopefully about a 10-minute block of time for questions from the group for any of the presenters. So we'll reserve those for the end of each cluster of presentations. You'll all share your own screens and advance your own slides yourselves. And then please do introduce yourself, your institution, your background, clinical, scientific, academic, whatever it may be, so we can all get to know each other better. So that's the housekeeping notes here for session 1. And as you'll see, we'll follow the agenda, and we'll start with Mariam Mughali here at Yale, who will talk to us about her work on ethical AI-enabled medical care. So Mariam, the floor is yours.
Thank you so much, Jason. So I'm going to share my screen. Can you all see my slides? Awesome, thank you. My name is Mariam Mughali. Regarding my educational background, I did my medical degree in Iran, my home country, and then moved to Canada and did the master's degree in e-health. And I came to the US for my postdoc position. And currently I'm a postdoc at Yale School of Medicine. Today, I'm going to present the work that I've done with a great team listed here on identifying the barriers and facilitators to trustworthy and ethical AI-enabled medical care. So I'll start by a quick background. As you probably know, artificial intelligence, AI, and machine learning are commonly used in healthcare to improve the prevention, diagnosis, treatment, and maintenance of healthcare conditions. More than 95% of the healthcare organizations in the US either already have an AI strategy or are planning one. However, patients and healthcare providers still are concerned about the trustworthy and ethical use of AI/ML-based software in medical care. And if they do not sufficiently trust the AI, they probably will be reluctant to use it, which could eventually lead to missed opportunities for improved healthcare outcomes. Existing research has begun to explore patient and clinician perspective on the use of AI/ML-based medical devices in healthcare. However, data about ethical concerns and trust barriers and facilitators are still sparse and mainly unactionable. Accordingly, we developed our research question that when using AI or ML-based medical device in healthcare, first, what are the main trust barriers and facilitators? And second, what are the ethical concerns and potential mitigation strategies? And for the purpose of this presentation today, because of the time limit, I will only focus on the second part of our research question. To answer our research question, we conducted a rapid review of literature through searching 6 databases. And the screening was done by 2 independent reviewers. Next, we conducted citation chasing from the included studies to make sure that we captured all the relevant information in the literature. And finally, we extracted the relevant outcomes. For the study selection, we had certain inclusion and exclusion criteria. Our focus was on the trust, transparency, or ethical concerns and facilitators from patients, caregivers, or healthcare provider perspective when using AI/ML-based medical devices in cardiovascular care. And we focused on cardiovascular because of the high number of AI/ML-based devices authorized for use or under review by the FDA in the cardiovascular care. And we included all studies published as full manuscript, and we did not set a time limit for the publication dates. Next, we excluded conference abstracts and book chapters, animal studies, and non-English publications. The treatment diagram here summarizes the different phases of our study. After running the search, we identified more than 10,000 papers, of which around 8,000 were unique. And after reviewing the abstract and title of those 8,000, we identified potentially 126 potentially relevant papers. And after reviewing the full text, we included 55 of those. And next, we did citation chasing, and we found an additional around 90 papers. And since we found that we reached data saturation, we stopped further rounds of citation chasing. And in total, we included 145 papers in this analysis. All the 145 papers were published from 2014 to 2022, except for one which was published in 1996. And the majority of these papers were review articles, but we also found 32 commentaries, editorials, or perspective pieces, 22 original research papers, and 3 case studies. And the majority of papers, around 85%, did not specify the indications for use for the AI or ML-based devices. And among those that specified, arrhythmia and heart failure were the most commonly indications. So after reviewing these papers, we found some ethical concerns from patient and healthcare provider perspectives, and unfortunately, we couldn't find anything from caregivers. And we classified them into 6 categories. And we also found some mitigation strategies for some of these concerns. So starting from privacy and confidentiality, there were some concerns around the potential misuse of personal information, issues with data storage, security or cyber attack, data sharing, or insufficient data de-identification. And we found that if the data is de-identified or anonymized, and if the security of the system is improved, it could mitigate some of the ethical concerns here. And next, for the inequity and bias, there were some concerns around suboptimal performance of AI tools in healthcare for certain patient groups, especially in the minorities. And also some were concerned that AI tools could be used to improperly profile patient. And also there might be an unfair distribution of benefits and burden when using AI tools in cardiovascular care. To address these, we found that it's important to make sure that the data that is used by the AI tools is diverse enough to be representative of the overall population. And sometimes it might be necessary to use specified algorithms for a certain population. Next, we found that it's important to engage different stakeholders, especially minorities, in different phases of developing and using the AI tools to make sure that all the voices are heard. And also, it's important to make sure that AI tools are available in under-resourced areas. The third concern is about patient harm, especially because of the potentially inaccurate data or the flawed algorithm that could be used in the AI tools. And also sometimes the decision support tool could be unvalidated, or they can have unchecked decision-making and action. And some also were concerned that sometimes profit is prioritized over care, which can lead to patient harm. And we found that sufficient training to all the users, including patients, caregivers, and healthcare providers, could help to minimize patient harm. Also, it's important to rigorously validate the AI tools and conduct evidence-based assessment. The fourth concern was informed consent and autonomy, especially because of the lack of transparency about the AI tools. Obtaining consent might not be always feasible. And there's also another challenge about consent in data sharing, which is concerning for some groups. And finally, if the patient desired to withdraw consent later, it could be challenging when the care is provided through an AI-based medical device. And to mitigate some of these concerns, it's important to provide sufficient and clear information to patients as much as possible. So although many believe that AI is a black box, it's important to explain the information clearly to patients as much as possible so that they can make an informed decision about their healthcare. The fifth concern was about the accountability and responsibility, and that's tricky because of the involvement of different groups in development and use of AI, from groups from developers, hospitals, and healthcare providers, and others. And to address these, we found that if transparency is improved, this issue can be addressed, because improving transparency helps that when someone when there is a question about accountability, it would be easier to answer those when the reasons that lead to an action or decision is understandable. And finally, the last concern is data ownership. Who owns the data, who profits from the data? And we found that it's important to prepare models of data ownership in advance so that the regulations about these are clear. Interestingly, we found that establishing regulatory standard is something that can address all the identified ethical concerns, and papers focus on the need for a regulatory and legal system that oversee the use of AI and consider the implications and responsibilities for developers, patients, and healthcare providers when using AI-based tool in medical care, especially in cardiovascular care. So the project is still ongoing, but these are some conclusions that we made so far. We found that numerous barriers exist to trustworthy and ethical use of AI/ML-based medical devices in cardiovascular care, and we think that further oversight by regulatory body could address many of these barriers and concerns, and we hope that these findings could inform labeling of AI or ML-based medical devices by the FDA. Thank you so much for listening to my presentation, and I will take any questions in the Q&A time.
Thanks so much, Mariam. Interesting and timely and important work. Again, if folks have questions, please jot them down. Keep them on your radars, and we'll have time at the end of this next batch of this first batch of presentations. So next, we'll hear from Susmitha Wanava from Harvard about natural language processing and medical device safety surveillance. So the floor is yours.
Thank you, Jason. Can you see my screen?
We can.
Good afternoon. My name is Susmitha Wanava. I'm a research fellow at the Harvard-MIT Center for Regulatory Science. I have a PhD in data science. My background is in machine learning and natural language processing. At the center, I'm involved in projects, many projects, but one of the projects that I'm involved in is regarding developing analytical tools for predicting medical device safety from both pre-market and post-market databases using natural language processing methods. And I'm very delighted to be here to present my work. And today my talk will be on one of our projects on NLP projects on creating an NLP benchmark data set for device safety surveillance. I worked with a team, a great team, and it's a collaborative effort. A background into NLP for medical product safety. NLP for in the drug space, especially for pharmacovigilance, has been a very active field and has seen many open challenges. NLP shit has, as you can see here, which have resulted in many curated NLP data sets for developing methods, research tasks, many research tasks, including identifying important pieces of information, relating it to drugs, and also conducting any classification on the reports or the clinical narratives. And also towards like the state-of-the-art techniques have used starting from rule-based systems to more sophisticated deep learning systems. However, on the device side, we haven't had any challenges or open shade tests, and we have none. We have no curated data sets that we can use to develop NLP approaches on. The shade tests that were conducted before are very limited and have used mostly rule-based systems. Rule-based systems are very effective, but they cannot be generalized to many other adverse events or other entity types. We have the goal here is to use more generalizable techniques for device safety. So today we'll just focus on medical devices, and as we see here, FDA adopts a total product lifecycle approach for medical devices, putting an emphasis on the assessment of devices from concept to post-market, and because in the pre-market, on the pre-market side where clinical studies happen and clinical evidence is gathered, we often don't have a lot of don't have enough time, cost, or enrollment to capture the long-term adverse events, and hence post-marketing studies and medical device reporting is very crucial. I think the Institute of Medicine recommends having a device surveillance system in place just because we have concerns of the rigor of the pre-market data due to many issues. And we have seen in recently there have been high-profile cases related to device injuries. So we can definitely utilize post-market data once the device is marketed. And another side of it is to use any healthcare data like electronic health records. However, the UDI, unique device identifier system, is being is in process, and we are not yet there where we can connect electronic health records fully to the adverse events. So there are multiple streams of real-world evidence when it comes to post-marketing surveillance of devices, from medical device reporting of adverse events, post-market surveillance studies, scientific literature, through which we can conduct these the surveillance. But today we will focus on medical device reporting for our project. The medical device adverse event reports are housed in FDA's MOD database. MOD data, the reports are submitted through the MedWatch forms. It's both mandatory reporting for manufacturers, but also voluntary reporting for healthcare professionals or consumers of the devices. As we see here, the number of reports have been increasing over time, with over 70,000 reports submitted every month. Studies have showed that primary source of safety signals triggered the medical device safety communications were from the direct reports or the real-world adverse reports to the FDA. So as I mentioned, MedWatch forms are used to submit these reports, and the forms have both structured meta fields and also unstructured text fields. On the left, you're seeing a sample unstructured report narrative, and the yellow highlighted text represents the structured metadata that's entered in the form, but the blue highlighted text represents the details entered into this narrative, and if we list them, as you can see that there are many adverse events or patient problems reported in the narrative compared to the metadata field. So our motivation for creating this benchmark data set is, we take this, if you look at the report on the left-hand side, this is a sample report from MOD on the publicly accessible interface. The patient problem mentioned is ischemia. However, if you look at the event description, it contains a lot more details about the event. If we try to tag this manually, these are all like the device identifiers, device problems, the procedure that this device is used in, the adverse events, but also the indications for which this device is used for. A lot more detail can be obtained from the narrative. However, manually mapping these reports is inefficient, as we have seen the volume of the reports keeps increasing every year. So here's where NLP can help us identify key pieces of information from the narratives and augment the structured data. To further demonstrate, relying just on the structured data itself has limitations. Structured data can be inaccurate and less specific, but also sometimes miss important pieces of information. As you see on the first example here, the patient problem recorded was no code available, but if we read the unstructured report, a lot more information on the patient problems is mentioned. This is not, however, captured in the structured data. The other example is a device problem. The device operates differently than expected. That's the coded problem. But if we see that it's a detailed device problems are mentioned in the report. And the reports, the narratives can have more specific text on the adverse events. Here, the pain was the patient problem, but if you see, it's more specific what kind of pain. The pain coded in the patient problem structure data is at a high level, but in the narrative, we can often obtain more specific patient problem data. Coming to this is another example where the device problem was mentioned as break, but if the narrative shows where the break happened, and because the fiber tip was removed by the surgeon, and where the break is in the catheter. Incomplete, many times we see that the patient problems coded miss some of the events that are mentioned in the report. So this is where NLP can help us identify these problems from the unstructured narrative and add it to the structured data, help in the surveillance process. Coming to the dataset creation, we have used the publicly mod MDRs and annotated more than 500 of the MDR reports. And we had a good inter-annotator agreement, and we annotated for 9 entities that span across device and patient problems, but also treatments and procedures. So this data set, once we are still in the process of completing our annotation process, once we have it, the data set would support a suite of NLP tasks, ranging from identifying key pieces of information in the reports to classifying the reports. For example, we can classify the reports to look for level of severity of the adverse event, or if it is a procedure related adverse event or a device related adverse event. There are some examples. So just to recap, post-marketing surveillance of devices is very much needed because we cannot capture all adverse events in the pre-market stage, but also MAR database is an excellent data source that we can rely on. And I think the NLP approaches have shown a lot of promise in the drug space. So applying those to the device space can help us augment the current post-marketing surveillance approaches and initiatives. So for our project, we have identified 2 challenges for applying NLP in this space, and we are trying to. Our goal is to come up with a data set first to support and to share it with the scientific community to advance NLP approaches and kind of extract important information from the reports. I would like to thank everyone for listening to my project, but also like to thank my mentors, Dr. Timothy Miller and Dr. Florence Bourgeois, for helping me on this. Thank you.
Thank you. Super interesting work. I know how hard it is to pack all the work you all have been doing into these 10-minute bundles, but really interesting. So again, hold questions if you've got any for our bundled question session at the end. But next we'll turn to Evangelos Okonomou here at Yale, who will talk about machine learning and predictive enrichment in RCT. So the floor is yours.
Thank you so much. Let me just share my screen first. Let me see. Just 1 second.
We can see.
I believe you can see my screen now. Let me know if not. Well, thank you so much for the kind introduction. My name is Evangelos Okonomou. I'm one of the clinical fellows in cardiology here at the Yale School of Medicine. And today I will be presenting some early results from my ongoing project on the use of a machine learning phenomapic strategy for adaptive predictive enrichment in randomized controlled trials. Now, these are my disclosures. We all know that randomized controlled trials are the regulatory and scientific gold standard in generating clinical best practices. However, we also know that they cost both time and money. Now, in a recent analysis of the estimated costs of the pivotal clinical trials supporting the approval of new therapeutic agents by the FDA between 2015 and 2017, it was estimated that the average cost of each trial was $48 million, with a median cost of $41,000 per each patient enrolled. And perhaps not surprisingly, the largest single factor contributing to that cost was the number of patients that was required to establish the treatment effects. This begs the question, what can be done to improve the design of clinical trials in such a way that we can minimize costs and maximize patient efficiency and safety? Now, in the traditional so-called fixed design clinical trials, participants are followed throughout the study period without any changes in any aspects of the recruited population. Now, on the other hand, the so-called adaptive clinical trials allow for prospectively planned modifications to one or more aspects of the design of the trial while data accumulate from subjects that are being enrolled in the trial. For instance, one can revise the power calculations at an interim analysis time point to ensure adequate power in the final study based on the actual event rates that are observed in the actual recruited population. Alternatively, one may dig a bit deeper into the accumulating data and identify which individuals are more likely to respond to the studied intervention. And this may allow the trial to continue by focusing on that population that is more likely to benefit, possibly also decreasing the required sample size of the overall trial. However, the challenge is, how do we do that in a way that is unbiased and defined a priori as part of the study protocol? Now, the idea of adaptive trial designs has recently emerged, and the FDA has actually embraced the need for regulatory innovation in this space. And this is best highlighted in this document and guidance that was published in, I believe, 2019 on the use of adaptive clinical trials for drugs and biologics. Now, in our recent work in the cardiovascular data science lab at Yale, we have been developing a phenomapping-based method that defines phenotypic embeddings of a clinical trial population, and this enables the discovery of distinct phenotypic signatures that are associated with differential benefit or harm from a studied intervention, and we have successfully applied this tool in a series of large cardiovascular outcomes trials. Now, building on this work, the specific objective of this project was to evaluate a machine learning guided adaptive approach that uses study arm effect differences for similar patients that are mapped based on their complex phenotypic features to design predictively enriched clinical trials. Now, this was a post-hoc simulation study based on real-world clinical trial data. So we collected data from two large cardiovascular outcomes trials, one of a drug and one of a therapeutic strategy. Now, in the first trial, the IRIS trial, almost 4,000 patients with a prior stroke or a TIA and insulin resistance were randomized to pioglitazone, a medication, or placebo, and followed for a median period of 4.7 years for the primary outcome of fatal or non-fatal stroke or myocardial infarction. Now, this study was published in 2016. We also analyzed the second study, the SPRING trial, where 9,361 patients with hypertension were randomized to more conservative or more intensive systolic blood pressure reduction, and again, were followed over a median period of 3.2 years for the primary outcome, a composite outcome of major adverse cardiovascular events. Now, both of these studies were completed in the past, and they both showed a significant benefit for the active intervention compared to the control arm. Now, in our simulation, we assumed a group sequential trial design. That means that we predefined 3 interim analyses once 50, 100, and 150 primary outcome events have been observed in each trial. And this allowed the trial to generate preliminary observations on relative efficacy and benefit at each time point that we could use to inform the next stage of each analysis. So how did we do that? So we designed the following algorithm. So we let each trial run until the first interim analysis time point. And then at that point, we paused and we create a phenomap of the trial population, observing the events that had accumulated up until that point. And then we explored the presence of heterogeneity in the population-wide efficacy of the intervention. If we did see signs of heterogeneous treatment effects, then we trained a parsimonious simplified model that could predict the relative benefit of an intervention as a function of a patient's pre-randomization baseline phenotypes. So we then repeated power calculations simulating event rates and the required sample sizes if we were to proceed with actual predictive enrichment based on our model for the next period of the study. if that was if all those criteria were met and we felt that we could actually predictably enrich the trial and maintain sufficient power with a smaller sample size, then we proceeded with predictive enrichment, where we actually selectively enrolled patients who were more likely to benefit. So we actually conditioned the probability of a patient being enrolled in the study during the subsequent period on their predicted benefit. Still, randomization was maintained. So each patient had an equal probability of being randomized to arm 1 or arm 2. However, if none of these criteria were met, we then resumed the trial and just let it proceed without predictive enrichment. And this cycle continued until the study ended. Now, although fully automated, in order to avoid any investigator input and therefore bias, our models are explainable. And we can, for example, see how the model develops as we proceed through the different interim analysis time points. So we applied this approach across 5 simulations of each trial, and we also ran a negative control where we randomly shuffled the covariates of the population in order to eliminate any actual heterogeneous treatment effects. So in the first study, in the IRIS study, we found that our machine learning-driven predictive enrichment approach resulted in an almost 20% reduction in the trial size compared to the original trial or the negative control simulation. And as expected, there was a numerically greater effect size for pioglitazone in the final population of the adaptive arm versus the original trial. And all simulations in the adaptive arm showed a significant benefit with a p-value which was consistently much lower than the alpha level of 0.025. Similar findings were seen in SPRINT, and here the method was able to reduce the final study population size by as much as 28%. We explored the nature of the phenotypic shifts with predictive enrichment, and we saw that actually our predictive enrichment did not impact the balance of the 2 randomly assigned treatments as expected. And there were actually slight changes or differences in some of the risk factors that could potentially explain the differential benefit that we saw in certain populations, but no specific population was actually excluded. We were also able to weigh the efficacy of the intervention against the potential risk at each time point. And by doing that, we were able to trend the net harm versus benefit for that population to make sure we were not selectively enriching for patients that were more likely to harm. And we do not see any evidence of that. Now, this is obviously work in progress, and our applications so far have been limited to post-hoc simulations of real-world data. The regulatory landscape is yet to be defined for trials with dynamic adaptive enrichment, but this is obviously work in progress and an area of interest. Integration of imaging and multi-omic biomarkers may actually in the future enable deeper phenotyping of ongoing randomized controlled trials. So in summary, what we propose here is a machine learning guided algorithm for the adaptive predictive enrichment of randomized controlled trials based on individualized treatment effect signatures from computational trial phenomaps. Our automated strategy can be defined a priori as part of the protocol design, requires no investigator input, and thus minimizes human bias. It's adaptive, it's stochastic, and it actually enriches for the relative benefit rather than the absolute risk of a patient. So ultimately, our proposed method aims to maximize the efficiency and safety of modern randomized controlled trials. I would like to thank all of you for your attention. The Yale-Mayo CERSI for funding this project, and my mentor, Rohan Kira, and all of our co-mentors and collaborators that have contributed to this project. Thank you.
Thank you. And again, questions will come just in a few minutes now after our 4th and final presentation in this first session from Arnaldo Suba Garcia on regulatory frameworks for EHR. So Arnaldo, the floor is yours, and your sharing is already visible. But you are muted and off camera if that's still in progress. There we go.
I'm sorry. The camera seems to be not acting. It's acting up.
That's quite all right.
Okay.
But we can hear you.
Good afternoon. I'm Arnaldo Garcia. I'm a practicing physical therapist, and I work for Baptist Medical Center here in Jacksonville. And I'm a CERSI Scholar at Johns Hopkins University. And today I will talk about why is it important to consider implementing a risk-based regulatory framework for electronic medical records. There are 6 situations that are important to bring into discussion to ensure that available electronic health record products in the market are safe and effective for their intended use. As many of you are aware, the clinical utility and bench utility of electronic health record has progressively been getting more powerful and useful, but comes with it are dangers and pitfalls that can result to patient harm. In this study, I have identified 6 of them in this research, and I'd like to share them with all of you today. These are, in spite of progressive evolution to a more powerful device, electronic health record continues to not be classified as a medical device. They continue to enter the market without market authorization. Second, the lack of pre-market review have resulted to proliferation of substandard product with poor usability. Dr. Melnick from Mayo Clinic have documented in 2019, using standard metric system usability scale, that the usability of the presently marketed electronic health records was consistently graded F by clinicians, with a mean SUS of 45. These translate to creating a 2:1 ratio of documentation per 1 hour of clinical care among clinicians. And per RAND Corporation study in 2020, the time burden of electronic health record documentation has also been correlated with a strong dose relationship to clinician burnout. Third, is there a widespread evidence based on court filings, recent court filings, that current EHR has led to patient harm and potential breach of privacy, and thus need a more comprehensive regulatory oversight. In a study done by the Doctors Company, a medical malpractice insurer, electronic health record-related lawsuits are on the uptick. Civil cases have risen from a low of 7 per year in 2010. This grew to 22.5 cases per year since 2017. Fourth, there is a widespread dissatisfaction among primary end users. The Global Harvard Health Institute in 2021 study estimated that clinician burnout related to the adoption of electronic health record is as high as 78%, calling it a public health crisis. In addition, peak among these topics are the emergence of AI, machine learning-powered electronic health devices and sensors with increased functionalities and power, enabling it to automatically participate in patient care. Examples of these are Abbott iStat and WellDoc's Diabetes Manager. These plug-in and play sensors are FDA-regulated medical devices. Taking this development deeper requires us to reconsider the vital role of the integrator, the electronic health record, in the management of patient, as it now is able to directly participate in the diagnosis, cure, mitigation, treatment, prevention of the human medical condition, passing the definition of medical device as stated in 21 U.S.C. 321, Section 201 of the Food, Drug, and Cosmetic Act. Finally, of great significance to all of us involved in clinical research is the leveraging of electronic health record for real-world evidence in pragmatic clinical trials allowed by the 21st Century Cures Act. Clinical trial sponsors have shown significant enthusiasm about this mandate, as shown by the increased number of registered pragmatic clinical trials. There are about 1,155 presently listed at clinicaltrials.gov. These clinical trials leverage big data collected from electronic health records and medical product registries to support an intervention's therapeutic claim of comparative effectiveness. A recently conducted study by researchers based at Duke University found that only a fourth of researchers performed fitness for use assessment correctly. Real-world data is not...
Arnaldo, I'm very sorry to interrupt, but we're still seeing slide 2, the background slide on your presentation. Have you been meaning to move along?
No.
Okay,
yeah,
just to be sure.
It's really actually still on the leveraging of electronic.
No worries. Just wanted to clarify. Go. I'm sorry to interrupt.
Just like what I said, a recently conducted study by researchers based at Duke University found that only a fourth of researchers perform fitness for use assessment correctly. Real-world data is not comprehensively examined prior to the use, resulting in changes down the line that are poorly controlled. In our lawsuit happy society, this is a recipe for poor science and future litigations. The objective of this study is to review available evidence for the impact of electronic health record on patient safety and its effectiveness for intended use, to identify gaps in current knowledge and design intervention for implementing a risk-based regulatory framework for electronic health record. The research methodology used in this research is the STARLIGHT. The study included case report, evaluation studies, government publication, legal cases, legislation, reviews, scientific integrity reviews, and systematic reviews in the last 12 years to examine the relationship between patient safety and electronic health record. The results eliminated duplicate and those of patients that were not relevant to the topic of interest. My search yielded 1,163 articles, of which 35 were included in this report. Effectiveness and safety for intended use themes included issues in the following areas. For the purpose of brevity, I will just focus on fraud and unequal burden of legal liabilities, as we have touched on the others in the previous pages. Fraud. In 2015, 31 medical societies have written the National Coordinator of NTC to express their concern that electronic health record developers are targeting function that meet government incentive requirement, rather than improving function vital to risk reduction and patient safety. Federal prosecution that involve mostly false claim involving robustness of electronic health records are uptick also, and have included major players in this field, including recent settlement from Athena, KPDP, Practice Fusion, and Greenway, with many other leading players in the process of investigation. And then second, but most important, is the unequal burden of legal responsibility. In this space, there are 3 legal practices that effectively prevent electronic health record to evolve into a more safe and effective product. These are the hold harmless contract, all end user must sign, the trade secret contract that they have to accept as condition of use, and the practice of learned intermediary concept. Vendors and manufacturers of electronic health records are successful in keeping their business private through a combination of these legal strategies. The hold harmless clause when signing contract with physician groups and hospital result in a unshared accountability for product performance. The trade secret for individual clinician effectively create a barrier for reporting of problems, potential risks, potential for harm, undercutting product surveillance. And the learned intermediary that states that a manufacturer of a product has fulfilled its duty of care when it provides all the necessary information to a learned intermediary who then interacts with the product. Liability is therefore shifted primarily to the doctors, the end users. This discourages sharing, reporting of patient safety risk by end user. So how do we get here? The infrastructure we have envisioned for electronic health record is that it will result to savings, evidence-based practice, and data-driven decision-making in medicine. So far, all of our investment and the 31 billion incentive we gave developers to contract this facility has resulted to burnout clinicians, poor interoperability, delayed maturation of the infrastructure, and patient harm. A quick revisit of laws that were passed to create this infrastructure allow us to see that the push to standardize the formats of electronic health record was realized with the passage of the High-Tech Act of 2009. The health information was enacted with the goal of facilitating the adoption of electronic health records with functionalities that improve healthcare quality, safety, and efficiency, and patient participation, privacy, and security, otherwise known as the meaningful use. What was interesting is that Congress revisited the regulation and electronic health record in 2012, and through the FDA Safety Innovation Act has empowered the FDA to develop a risk-based regulatory framework for electronic health record in tandem with ONC and FCC to encourage innovation, protect patient safety, and avoid regulatory duplication. Finally, another key legislation that is hoped to improve this problem was passed in 2016. Congress passed the 21st Century Cures Act to streamline pharmaceutical and medical device regulatory process to speed up their market availability. The Act also added interoperability requirements for and the ability to use real-world data and real-world evidence in pragmatic clinical studies. In this page, in my analysis, the absence of systemic regulation, full review, or market authorization mechanism has led to a deferred desire by developers to develop a better product. Because of this and the lack of assurance and safety and effectiveness verified through a rigorous testing, the use of these products poses risk to clinician, patient, and the public. It is important to note that medical societies have been vocal and vehemently and strongly critical of the current certification process, with a strong emphasis on the misalignment of end-to-end testing focus on hitting requirements for incentive, not on usability.
Ronaldo, I'm wondering if you could move, just for the sake of time, move to your.
I'm very close.
Thank you very much.
All right. The current state of affairs of electronic health record has resulted to patient harm and risk for public safety. And my conclusions is that the current state of EHRs available in the market compromise both patient safety and healthcare quality. In offering my other conclusions, I tried to be as objective as possible and wanted this research to inspire others to follow through to improve safety, effectiveness of product in the market, and public health. Thank you for listening. The next 2 pages are my reference for this study. Thank you.
Thank you very much, Ronaldo. Thanks to all 4 of our presenters. So we do have some minutes remaining for questions from the floor, all of which, it occurs to me, these talks give us insights into new and existing or potential tools and ways in which they can be structured or organized or in fact regulated to improve their use, their experience for patients, for providers, for regulators. So lots of commonalities even in the different domains we've explored here. So the floor is open if folks just want to use the raise hand function or even unmute. I think I can see everyone. Would love to hear questions from any of our 4 excellent presenters in this first session.
Fantastic work. Go ahead.
Thank you. No, Molly, please go right ahead.
Yes. So fantastic work, everyone. So for Dr. Wanava, I'm sorry if I got your name wrong. I'm really curious about some of the ideas of bias that Dr. Magali was talking about in her talk, and kind of how that feeds into the source of the data that you're looking at in MAUDE. One of the ones that you highlighted was like specifically from an attorney. Like, how do you think about doing that, or is that not really an issue in the kind of work that you're doing?
That's a great question. Thank you. That definitely is an issue. So with MAUDE data being like about 95% of them being from manufacturers, and those reports can be submitted through attorneys, lawyers. So there's a lot of variance in the structure of the report, the language that's being used. So we're actually looking at, in another project, the difference between the text that these different reporting sources use, and that definitely is showing that we have to take bias from the reporters into consideration. So with the models that we will use, we will definitely try to act on those biases and try to minimize them as much as possible before we start training these models.
Fantastic. Thank you.
I see Dan Keller has his hand raised. Please, Dan.
Thanks, Jason. Arnaldo, just a question for you, please. You thought your presentation there was very good, and something I didn't think about. I didn't think of regulating electronic health records, but it makes sense. And I'm just thinking now of the companies that have consumer electronic health records. So will the FDA need to do regulation on direct-to-consumer advertising from these companies who are targeting consumers for the uptake of their electronic health records?
I believe so. Thank you for your question. I believe so. I think, it's high time because of the widespread all stakeholder, effect of electronic health records. Meaning to say, like everybody's affected, for standardization and at the same time for safety and effectiveness. I think, the FDA, the FDA has a power to regulate electronic health records using the 2012, Pedestrian Act, but they haven't done so. I think, it is badly needed.
Thank you.
Other questions, Joe? I see you've unmuted.
Well, again, this is really a sort of a comment for Arnaldo. I don't know if you saw Greg Pappas, who used to be at CDRH, put a comment in the chat noting that ONC is looking into this, but they interpret the authority as not being able to regulate the safety and effectiveness of the medical records. And so I wonder if there's a non-patient harm related angle to the regulation. And of course, I'm also thinking about Susmitha's presentation and around thinking about NLP for adverse events, and could that be the sort of angle of the regulation, thinking about how the electronic health record can better understand medical product safety as opposed to the safety of the system itself. So I'm just curious what your thoughts are.
Yeah, my thoughts are initially, electronic health records are payer imposed to providers, meaning, payer really wants to, to have that documentation about the care that they are providing, that providers are providing to their members. But how do you call this? For safety and effectiveness, truly the Innovation and Safety Act of 2012 has given the FDA, a leeway to draw down regulation together with ONC and FCC to ensure that the infrastructure is safe and effective. I hope I answer your question.
And we're seeing lots of great connections being made in the chat to keep these conversations going, which is fantastic. A question in our last moment or two for Dr. Economou, forgive me. I don't spend a lot of time in the clinical trial regulatory landscape world, but my sense is there's a certain conservatism about approaches in this area. What is your sense from thinking about these areas, talking with folks about, is there an appetite, is there an openness for these kinds of innovation, or is that going to be a large hill to climb to implement the kinds of strategies you're thinking about?
I think that's a great question. I think there's definitely an appetite, there's definitely an interest, and we've seen that in our early discussions with some key stakeholders. I think there's some sort of reluctancy in because even though the need for innovation in the adaptive trial design space has been highlighted in the past, there's been some reluctancy in adopting some of those methods in actual clinical trials, and mostly because I think the regulatory landscape is still a bit, hasn't been clearly defined yet. And there's been ongoing work on how can we optimize the design of those trials to ensure that they are safe for the patients and they are actually gonna provide enough sufficient evidence to justify the approval of a potential therapy or drug or device. So there's definitely an interest, and I think that's why it's a great area for research. But I think ultimately the regulatory landscape is important to be determined so that we can start applying those trial designs in actual trials.
Thank you. Any final questions for this first group before we move ahead to our next session? Seeing none, let me thank all four of our presenters. A great kickoff to the afternoon. Lots of great things to think about and more to come for sure. So I think we're moving right along to session 2, keeping a bit of our 10-minute head start. And I'll turn things over to Molly, who will lead this next session.
Wonderful. Thank you, Jason. So I'm not going to take a ton of time introducing the panel, but we have a fantastic group of 5 abstracts that we're going to hear from scholars at Mayo Clinic, UCSF, Yale, and Johns Hopkins. So we're going to go in the order that's in the program and start with Dr. Kelleher.
Great, thanks, Molly. Hi everyone, my name is Dan Keller. I am a research associate in health economics at Mayo Clinic, and I have a PhD in health economics from the University of Galway in Ireland, and I conducted my PhD research with the Euroqual Research Foundation in the Netherlands. My PhD research largely focused on looking at health preferences and eliciting and modeling health preferences. So since at Mayo, I've been able to work on a couple of studies looking at preferences for different healthcare interventions or healthcare practices. So the title of this talk is Examining the Preferences of Adults with Depression on Psychotropic Polypharmacy and Healthcare Providers for Pharmacogenomic Testing at Mayo Clinic. And just a quick background. So over 50% of adults with depression in the US are on psychotropic polypharmacy, and that's taken from Professor Rosenheck, who is a professor at Yale. And some of the individuals, so of these individuals, they may be at increased risk of adverse drug reactions due to the drug combinations. And for example, that may be individuals with depression who are being treated with serotonin drugs may be at risk of developing serotonin syndrome. So these individuals, it is imperative that they are able to access treatments that are safe and effective and that minimize the risk of adverse drug reactions. So as such, some individuals may benefit from the use of pharmacogenomic testing before being prescribed antidepressants and other pharmacological treatments to enhance potential treatment response and minimize the risk of developing ADRs. So PGX testing examines how individual inherent generic variations affect drug absorption, distribution, metabolism, and can be used to predict response variability for certain drugs, which can facilitate individualized disease management and reduce ADRs and improve health outcomes. That was taken from Dr. Zara's paper, who's presenting actually after me. So the objectives of the study, so while there may be clear clinical utility associated with PGX testing for individuals, understanding what factors encourage or discourage the uptake of such tests is important to ensure the level of uptake is sufficient. So the 2 main objectives are to examine preferences of patients with depression and psychotropic polypharmacy. Again, it's very important to understand what are the issues or what are the factors that patients do care most about when looking for these sort of type of tests, and to examine the preferences of healthcare providers who may be treating patients with depression and psychotropic polypharmacy for the same test, to see if these are the gatekeepers to such tests, what factors are important to them when they're looking to order tests on behalf of patients. And just to give you a quick overview of the methods, and I should say, sorry, before I go on to this study has not yet started. We are in the process of putting together an R21 grant to fund the study. So it looks like the study does need about 250,000 in funding, sort of looking hopefully get this funding application in. Hopefully then the study can start not before too long. So it will be a mixed methods approach. So when you're looking to understand patient preferences or healthcare provider preferences, it is best to understand first what are general overall attributes of a healthcare intervention or healthcare practice that are most important to the group you are looking to study. So to do so, we're going to first conduct nominal group technique, focus group discussions with about 30 patients and 30 healthcare providers, keep them separate, have 2 separate groups, a patient group, healthcare provider group, split each group into about 5 to 6 different groups, conduct these focus group discussions to get a better understanding as to which kind of factors associated with a PGX test would these 2 groups be most interested in, and what do they think is most important. From that, then that level of information, that qualitative information, informs our design of a discrete choice experiment study. And it pretty much sets us up with 2 types of data, which is great, and 2 separate studies within this one study. And then we're looking to we're recruiting patients from the 3 Mayo Clinic sites, Rochester, Florida, and Arizona. And we've got a team of researchers in each hospital. So I should say actually the rest of the team members are Drs. Bijan Bora, Dr. Jewis Samatter, Dr. William Bobo, Dr. Isa Hoink, and Dr. Shihad Nazri. So we have a great mix of team of doctors across the 3 sites, because then we're looking to then recruit the doctors in. We're looking to get about 200 healthcare providers. It's obviously best to have champions in each of the hospital to hopefully increase our survey response. And then for the patients, we're looking to recruit about 400 to 500 patients as well. And patients will be recruited, those who have a diagnosis or of depression, and are also on psychotropic polypharmacy, which is individuals who be taking 2 or more psychotropic medications. And here's just an example of what PGX testing attributes could look like. So this is taken from Chen et al. And they looked at patients who were on polypharmacy and looked at their preferences for a PGX test. Now, they did not look at patients on depression. They looked at patients with psychotropic polypharmacy. But they broke down the PGX testing attributes to look at identifying the best medication and dose, identify, prevent side effects, privacy access, involvement and advocacy for my treatment, communication of test results, and cost insurance. And you see on the right side there, the box, then each attribute has associated levels, levels ranging from 2 to 3 levels. And then this is an example of a choice task that an individual will be asked to respond to. So we're hoping that we're going to have about 10 to 12 responses. And 12 have choice tasks. So you can see the attributes are be asked to pick which is better, test A or test B. Each test is described using the attributes on the left-hand side, with then varying levels associated with each attribute to describe each test. From that level of information, then we're able to model the preferences of each group, and ideally we'll be getting the healthcare provider group and the patient group to respond to and value the same set of choice sets. So just the impact of this research that I think will come from it. So there is practice and policy implications in the sense that from Mayo Clinic itself, it's, as I said at the start, it is important to understand the factors that encourage or discourage the uptake of such tests from the point of view of the patient, from the point of view of the healthcare provider, and seeing how they compare. Having understanding of this type of research allows us to then be able to predict the potential uptake of such tests, and that information can then inform policy initiatives to inform the service provision of such tests at Mayo Clinic. Minimum of 2 papers will be published, since we have this NGT, the nominal group technique focus group data. That will be a qualitative paper, hopefully published in a more specific journal related to personalized medicine or pharmacogenomics. And the second paper, looking at the DCE data, maybe look to be published in a more specific health economics journal. And there's potential to inform regulatory decision-making from this research, especially in the case of where we're looking at to examine healthcare provider preferences, and in the instances where these healthcare providers are the gatekeepers who are ordering such tests. So it is important to understand why they're ordering tests and what factors are very important to them. And great scope for future areas of research, in the sense that we're looking now at the preferences for this patient subgroup. There's a patient group for pharmacogenomic testing. This type of research could be extended to other patient groups in other clinical areas to examine what are their preferences for PGX testing, and same for healthcare providers. And then just how the study ties into the FDA CERSI priority areas. It primarily relates to priority area 3A, so scientifically valid approaches to incorporating patient preferences, as this study will capture patient and healthcare provider preferences that can be incorporated into regulatory decision-making, and ties into also priority area 1B, advanced development of individualized therapies, as it will advance knowledge of patient and healthcare provider preferences for PGX testing, which then can be used to improve or be used in the development of individualized therapies also. So that's that. Thank you for that.
Thank you so much, Dr. Keller. Next up is Dr. Zhu.
Okay, thank you, Molly. So can you see my screen now?
Yes, it's perfect.
Okay, thank you. Hello, everyone. My name is Julia, and I'm a research associate at the Knowledge Synthesis Unit at the Current Center. And my background is I have an MD from Peking Union Medical College in Beijing, China. And then I pursued a couple of graduate degrees here in the United States. And especially I got my PhD from the University of North Carolina at Chapel Hill in health policy and management. And now my major research interest is in health outcome evaluation and the clinical implementation of pharmacogenomics. So Dan and I, we share very close research interests, which is very nice. And I'm glad to hear your presentation. And you laid a very good background of pharmacogenomics for me. So I will keep simple on my background. That was nice. And I'm moving on to a pediatric residency this summer to University of Nevada at Las Vegas. So this project. So this is the background of my project, is the pharmacogenomics, of course. So this pharmacogenomics is a broad study area that investigates the gene variations, which will lead to drug responses differences in different people, and eventually will lead to different treatment outcomes. So by far, there's a lot of evidence that actually already exists in this area, this field, as Dan has mentioned about, especially in the psychiatric area, but actually it's in cardiovascular and in a lot of other therapeutic area as well. And it's found that pharmacogenomics can really prevent drug adverse events and also maximize drug effectiveness and improve cost effectiveness as well. So US FDA actually has a very nice 2 tables of the pharmacogenomic knowledge. One is the pharmacogenomic biomarker in drug labeling. Actually, those knowledge are included in the drug labeling, and by far it's 367 drugs, as I have screenshots here, and including 19 therapeutic areas. As you can see that the biggest area is oncology, and the second area is neurology, and the third area is psychiatric. So this study, as actually there's the frequency the frequency is the number of drugs, and also the drug labeling is actually very detailed information regarding the dosing of drugs, how to change the drug dosing regarding based on patients' genetic test results, or how to pick a different drug for the patients. So the next table is a table of pharmacogenetic associations, and they have 3 sections. It's very small here, but as you can see, I highlighted in dark blue here. The section 1 has 63 drugs, and those are the information has strong evidence of pharmacogenomic associations between the genes and between the drugs. So these are really nice knowledge resources for the pharmacogenomics in the US. Internationally, there is already a lot of societies, they are really interested in this area, and a lot of effort has been going on in this area. Like there's a CPIC, it's basically like, it's basically the Bible, or it's very it's very updated knowledge in this area. It's the Clinical Pharmacogenetics Implementation Consortium. And also the Dutch, Netherlands has a very nice Dutch pharmacogenetics working groups, and Canada is a little bit ahead of other countries as well, and they have the pharmacogenomics networks for drug safety. And the PharmGKB is the clinical guideline annotations. They actually is a very nice comprehensive knowledge source. They synthesize all kinds of resources of pharmacogenomics and the drugs and genes into this into this annotations. So given all those evidence and all those knowledge, we can see that pharmacogenomics can benefit the clinical practice a lot. But the thing is, by far, physician has a really critical role in the process of implementation. But by far, clinicians, physicians' attitude and opinions really is very diverse and discrete. So especially the knowledge, by far is very is by specialty, or it's only by specific knowledge. So we think it's critical to give a really broader view of how physicians really picture this area, how physicians picture this tool, which can help them in the clinical practice. So the study has 2 aims. One aim is to identify the physician's attitudes and opinion towards clinical use of pharmacogenomics. And the other thing is, if it's possible, we're going to explore different attitudes among different physician characteristics groups, like age group, year of practice, specialties, and so on, like gender. So the study method we're going to use is systematic review. So we are going to use multiple databases to look at this topic, and the time frame will be the past 10 years. Since during the past 10 years, it's not really a lot of knowledge has been generated, but the practice didn't really change a lot. So we're going to look at why this topic has stayed there, the clinical implementation stayed here during the past 10 years. And also we're looking at original studies and the systematic reviews. The population we're going to look at is physicians, medical doctors, and clinicians, and also with the specialties of primary care, general practice, that's what's called in European countries outside of the US or Australian countries. So family medicine and the 19 FDA drug labeling specialty, as I mentioned in the previous table, and also pain management. So the intervention we're going to look at is pharmacogenomics implementation. And also the outcome evaluation we're going to look at is the physician's attitude and opinions. So by far, we just started the project and we have some preliminary results, but it's very interesting. So we found 2,732 manuscripts from those multiple databases, and 111 manuscripts are pre-screened and the most relevant, and 86 studies eligible for study extraction. But this number is subject to change because we're still looking at other updated databases as well. So the results suggest that majority of the study actually is survey study, interview study, focus group, mixed methods, very, very small quantitative analysis. And some use survey interview, but it's all qualitative. Physician specialty is, there's a lot of family medicine, general practice, psychiatric, surgery, pediatric, internal medicine. But for other specialties, like I mentioned in the FDA, the study is actually very, not really rare, but relatively smaller number. So as we can see, PGX, that's pharmacogenomics, assisted interventions, we can see as the general therapeutics, very broader concept, or the medication choices, medication dosage, and the smoking cessation actually is a big one. And the drug side effects, of course, is a big domain as well. So overall attitudes, as I mentioned about, is very decreased. A lot of optimistic, supportive, the physician reserved, or even not supportive, or physician feel not prepared. So we can summarize those different opinions into 5 domains. So the treatment effectiveness domains is, as we can see, the pros, which means physicians, they're supportive of pharmacogenomics. They think this is provide confirmation of a clinical decision. So this is very interesting to see, because as we can see the attitude, physicians consider the pharmacogenomic knowledge as the supportive confirmation information instead of their own judgment. So this is very important to keep that in mind, I think. And also they can help predict the patient's response to the drug and influence the dosage, and also help pick alternatives to the current drug. The cons of physicians, if they think racial difference, for example, the racial difference in the genetic knowledge may lead to disparities in treatment effects. This is very interesting. I put it here because by far, a lot of clinical knowledge, especially in the pharmacogenomics area, a lot of knowledge generated from Caucasian and or Asian countries have a lot. And the minority like Hispanic or African-American, the database actually is relatively smaller. So if we generated that knowledge from those races, it will create actually inaccurate knowledge for those minority patients. So that's a very advanced cons from the physician opinion. And from the health economics domain, a lot of physicians think it will improve treatment cost effectiveness. And they support genetic tests should be reimbursed by the insurance companies. But at the same time, the major concern is the patients who with the test positive results, they will be disadvantaged by the health insurance. Insurance can cherry pick them. So in the physician-patient relationship domain, and a lot of physicians think they can improve the therapeutic relationship because they can incorporate it into the patient-physician conversation and then make the conversation more smooth and more convincing. And also they can build a level of trust, because patient trusts the physician because the patient thinks the physician is very knowledgeable and knows about them. And there is a lot of disadvantages, I think, is because the time is already constrained, very limited during the visit. So if you talk about pharmacogenomics, especially a lot of time, they have to go over all those consent forms. So it might not have enough information, enough time for that. And also some physicians think if you overemphasizing and relying on the genetic results, you might miss a lot of clinical characteristics of patients, so that it will actually misleading the whole diagnosis, and also it will provoke patient anxiety as well. So from the other, because the time is constrained, I think I'm not going over to the other those points, but there's a highlight is what physicians think if they physicians who's familiar with FDA tables, they actually feel more prepared versus those one they're not familiar with this knowledge, they feel they are not prepared. So this is indicating that we can use FDA the tables as a very important knowledge source for this patient, for the physician as well. So the clinical implementation of this project is because we already have the research findings from pharmacogenomics, we want to cross the chasm of this adoption and translation this knowledge into to improve the clinical outcomes. So in the CADM, that's the barriers. Of course, there's opportunity as well. So this project is to help identify what's in physician's mind. So how to incentivize or help them to cross this and eventually to pick up this tool in their clinical practice. So the policy implementation and implications of this project is, as we can see, this is from one of the book chapters I wrote, authored with my mentor, first mentor, Dr. Bora as well, which is who is Dr. Keller's mentor as well. So this is a figure about how the system works for pharmacogenetics using cardiovascular disease as an example. As we can see, all the healthcare systems around the patient, and the providers are the first layers around the patients. So while the payer or the institution, they are all supportive of this activity, clinical practice. So if we can identify the practitioners' opinions, attitudes, and all those layers will have a better way to help them facilitate the whole system and improve the system, how it works. Okay, thank you so much. This is the end of my presentation, and you're welcome to email me with any questions. And this is my mail email. And since I'm moving on to my new position this summer, so this is my new email as well. So thank you so much.
Thank you so much, Julia. That was fantastic. Congratulations. I had not heard your news.
Thank you.
Next up is Mr. Jason Davidson from UCSF.
Thank you. Let me pull up my slides. Share my screen.
Perfect.
Right. Hello, everyone. Happy Friday. My name is Jason Davidson. I'm a third-year PhD candidate at University of California, San Francisco in Dr. Atul Butte's lab. And today I will be talking to you about the evaluation of social determinants of health on type 2 diabetes treatment selection and disease severity. So first, let's get into what are social determinants of health. They are the conditions in the environments in which people are born, live, learn, work, play, and worship that affects a wide range of health, functioning, and quality of life outcomes and risks. More specifically, social determinants of health, hold on, my pointer. Social determinants of health involves information related to an individual's health, including healthcare access and quality, social and community context, and neighborhood and built environment at the population level. And at the population level, we must ask what social determinants of health factors are contributing to the overall health of a population. And individually, social determinants of health factors such as education, access, and quality, behavioral factors, genetic factors, psychosocial, and social demographic factors all contribute to the overall health of a patient at the individual level. And lately, clinicians have strongly noted that social determinants of health plays a role in clinical outcomes, but there has not been a way to clearly define these in clinical research. And because of this, it has not been widely adopted in real-world evidence studies yet. Most of this is partly due to the difficulties of measuring social determinants of health at the individual level, but luckily we have ways to measure this at the population level by way of census tract information. Oops. Here we go. Social determinants of health is extremely important because we recently have noticed that it plays a role in health risks and outcomes, especially with chronic illnesses and diseases. Because of this, we plan to model our study on type 2 diabetes because disparities in diabetes burden exist in a large part because of social determinants of health at the individual, subpopulation, and population level. Previous translational research and practice addressing health equity in diabetes have generally focused on changing individual behavior rather than targeting social determinants of health to increase health equity. Therefore, the next generation of diabetes translation requires health equity goals, consensus definitions, core measures, multi-sector partners, enhanced training, and expanded funding to prevent diabetes and treatment, and also to overall increase health equity. So the question for our study is, does social determinants of health impact treatment selection and type 2 diabetes health outcomes? But with our question, how exactly are we going to capture this data, and what databases are we using to answer these questions? Before we get deep into our study, let's talk about the University of California Health Data Warehouse, where it's a structured EHR database that we will be using for our study with 6 academic health centers, over 8 million patients that serves very different parts of California, both geographically and demographically, where de-identification of this data has already been completed to enable clinical research projects, and the data is stored in the OMAP vendor-neutral open data model, which enables a wide range of software tools and computational methods to be used within a community of more than 400 researchers across the UC health system. And early real-world evidence studies have improved our understanding of the treatment practice of type 2 diabetes, but the utilization and health outcomes of treatments does not represent social determinants of health, which has resulted in a lack of knowledge on which treatments are most effective and most widely distributed across distinct population groups. In this figure by a former lab member, he showed the differences between patients on monotherapies and combination therapies across the UC health system at all of the different UC health centers, which represents the patients changing treatments from their monotherapies to combination therapies to manage their type 2 diabetes type 2 diabetes disease progression. And this paper showed that we have data on the treatments of patients, but now it's time to take it a step further and determine the social determinants of health of patients and how does it ultimately affect treatment choice and outcomes. We plan to evaluate social determinants of health in the study by using the Area Deprivation Index, which is a multi-dimensional evaluation of a region's socioeconomic conditions that are linked to 17 different calculated census tract variables, which are categorized by education, housing, poverty, and employment status. And within our database, we have over 5 million patients with their ADI rankings. Now, ADI is important because it describes area-level social determinants of health characteristics that are mapped to each patient based on their zip code. And across the University of California Health Data Warehouse in the state of California, we see that there's a wide distribution between patients who are ranked with 1, meaning least disadvantaged, meaning that they live in richer, more well-off neighborhoods, and patients ranked in 10, which are more disadvantaged, meaning they live in poor neighborhoods. And across the state of California, we see that there's a wide variety of patients who are categorized in rich neighborhoods and or poor neighborhoods across the state, which makes our database the perfect database to use this study on. Now, ADI is important for the study because 1, it will be used for risk adjustment to improve predictive models at an institutional level, to identify high-risk patients and health disparities, and to design better care pathways that improve health outcomes across the board. So into our study design, for our cohort selection, we identified patients from our database with a single anti-hyperglycemic therapy. We then excluded patients with other type 2 diabetes medications before this index date, and we're left with patients with only their single-use orders. We then excluded patients with previous history of type 1 diabetes or any other secondary diabetes, which then gave us new users for each monotherapy. We were then left with we then excluded patients without a type 2 diabetes diagnosis before their index date to exclude patients who were given treatments for other means except for type 2 diabetes, which then left us with new type 2 diabetes users. And then we further excluded patients who did not have their hemoglobin A1C lab measurement within +7 or -7 days of the index date, which left us with the new users, along with the demographics and age, sex, race, and ADI. So into our baseline characteristics, we showed that in our data set, it was pretty split across the board with 52% male, 48% female. Distribution of patients by ethnicity, we showed that 68% were not Hispanic or Latino, 25% were Hispanic or Latino, 8% were unknown. The distribution of patients by race, we noticed that there's a higher rate of white people in our data set because that is also similar to the background of the population of the state of California. We then had 18.2% other, 14.8% Asian, 9.2% unknown, 7.1% Black or African American, 3% multi-race, 1% Native Hawaiian, and less than 1% American Indian or Alaskan Native. And what we also noticed that according to the census demographics of the state of California, we noticed that the minoritized races actually outweighs the normalized state percentages in this data set. Looking further at our characteristics, we wanted to look at the distribution of patients by ADI. And what we notice is that there's a broad distribution within ADI, but we also noticed that patients from richer neighborhoods, more well-off neighborhoods, actually had a higher percentage of data in our data set, whereas patients from poor neighborhoods had a lower percentage of data. And we wonder maybe that could contribute to a lack of healthcare access and a lack of healthcare affordability. Then looking at our patients by health center, we showed that UCD at 37%, UCI 19%, UCLA 17%, and so forth. But what was also interesting to notice was that according to the ADA guidelines, we know that metformin should have a higher use as the first monotherapy for patients. We noticed that 50% in this data set of patients had metformin, followed by 24% with sulfonylureas, 17% SGLT2 inhibitors, 3.9% DPP4, 2.8% GLP-1 agonists, 1.5% thiazolidinones, and less than 1% alpha glucose inhibitors. So then we ran a regression model. With our data set containing ADI and hemoglobin A1C data points, we performed an ordinal logistic regression model with ADI as our dependent variable to then understand the association of hemoglobin A1C for patients with different ADI. And what we notice is that with the patients from poor neighborhoods are actually having higher probability of receiving type 2 diabetes care at a greater disease severity, meaning with a higher hemoglobin A1C level than patients from richer neighborhoods. And as you see, when there's a lower disease disparity, it's completely flipped, where there's a higher probability of their patients from rich neighborhoods receiving their therapies at a lesser disease state than patients from poor neighborhoods. And this outcome truly raises the question as to why patients from poor neighborhoods are then to receive treatments at a much more severe disease severity than those from rich neighborhoods. And this is a question that we remain to have an answer and should all aim to answer to increase health equity in communities that truly need it the most. So in conclusion, there we go. In conclusion, we found that rich and poor neighborhoods receive care at different disease severities. And this study shows that we cannot ignore social determinants data if we truly want to help increase health equity And social determinants health also shows us that it can help advance clinical decision support tools for clinicians in the future. And I would like to acknowledge my lab and thank you. And if you have any questions, feel free to email me or just scan this QR code, and that has all my contact information. Thank you.
Thank you so much. That was a fantastic presentation. Really appreciate it. Next up, Mr. Sam Yoon from Yale.
Okay, great. Let me share my screen. All right, great.
Perfect.
So good afternoon, everyone. I'm Sam. I'm a medical student at Yale, and today I'm going to be talking about my project titled Evaluating the Evidence for Surrogate Endpoints in Chronic Diseases from the FDA's Table of Surrogate Endpoints. This project is still ongoing, so some of the results that I will present will be preliminary. Really, what the aim of the project is to understand what is evidence supports the use of surrogate endpoints that are used for the approval of novel therapeutics. So starting with some background, some definitions. So clinical outcomes directly measure whether people in a trial feel or function better or live longer. These are typically things like survival rates, incidence of cardiovascular events, cognitive function. On the other hand, surrogate endpoints do not directly measure clinical benefit, but rather are measures that are able to predict that clinical benefit. And so surrogate endpoints are used both in traditional approval and also in accelerated approval. And since the introduction of the accelerated approval program, over 200 products have been approved through the basis of surrogate endpoints. Some examples of surrogate endpoints are LDL cholesterol for occurrence of MI, forced expiratory volume in asthma, and famously HIV viral load in the development of AIDS and HIV. So in 2010, the Institute of Medicine released a report titled Evaluation of Biomarkers and Surrogate Endpoints in Chronic Disease. The goal of this report was to develop a general framework and way of thinking about how we evaluate biomarkers and surrogate endpoints. What they did was they proposed this three-step framework, the first being analytical validation. So these are characteristics that are intrinsic to the biomarker or surrogate itself, things like sensitivity, specificity. The second step in evaluating a biomarker would be looking at qualification. So what evidence is there that supports this particular surrogate's association with a meaningful clinical outcome? And the third was a step looking at utilization. So what's the context of use, whether it be diagnosis, for treatment, or for something else? And so since then, a lot of studies that look at the performance of surrogate endpoints have been largely in oncology. And so in oncology, we see a lot of drugs that have been approved based on surrogate endpoints such as ORR, which objective response rate, which looks at tumor shrinkage, or PFS. And in previous reviews, it's been found that the majority of correlations between surrogates and overall survival were of low or medium strength. In another review that looked specifically at breast cancer, they found that none of the correlations between surrogates, such as ORR and PFS, and overall survival were correlated except for one. So although we have a lot of reviews that have looked at how good surrogate endpoints are in oncology, there's no review that has really looked at the same strength of surrogate endpoints across a larger set of diseases, and that's what we wanted to do. So our starting point was a table published by the FDA. So they make a public list of surrogate endpoints that have been used as the basis for approval of a drug or biological product. And included in this table is the disease or use, the patient population, the surrogate endpoint, and the type of approval it's appropriate for, whether it be traditional or accelerated. So our question for this project is, what is the strength of association between surrogate markers and clinical endpoints in chronic diseases based on accumulated evidence? And the way we want to answer that question is through an umbrella review. So if you will, a systematic review of systematic reviews and meta-analyses looking at this association. So very broadly, our process was first determining which indications we want to include, searching for each of those indication and surrogate pairs, screening for reviews, evaluating evidence, and ultimately synthesizing everything together. So we started with the FDA table that I had just talked about. And in there, there's 124 disease and surrogate endpoint pairs. We specifically wanted to look at chronic diseases. So we excluded diseases that were genetic, vaccine-related, acute, or oncologic. Given that there's already been done a lot of research looking at cancer endpoints, we decided to focus on things that were non-cancer related. So that left us with 37 disease surrogate pairs. And so for each of those pairs, what we wanted to do is we wanted to conduct an individual search. And so we developed a search strategy. Initially, our search strategy was centered around 4 main concepts, the first being study design. So we had a set of search terms that specifically would pull papers that are systematic reviews, meta-analyses, pooled analyses. So that was our first search term. We then had a search term that was specifically what looked for surrogates. So these included terms like surrogate, intermediate, biomarker, and this was aimed to pull papers that specifically looked at that. Then we had a search term for correlation, and finally we had a search term that was for that specific indication or disease. What we found when we were doing this search is that we really kind of wanted to cast a broad net, and there were some cases where we were coming across papers where they didn't use surrogate-specific language. So they would examine, for example, the link between maybe blood pressure and something else, but they wouldn't use the vocabulary of surrogate. So what we ended up doing is we ended up doing another search to supplement our initial search, where we took out that surrogate language from the search and instead added a surrogate-specific term. So instead of having just surrogate intermediate biomarker, we would have the specific surrogate used for that disease indication. And we added that to our first search to make sure we really casted a broad net. So here's an example of our first search strategy. So you can see 1 through 6, these are terms that are meant to specifically pull out meta-analyses for systematic reviews. So all you need is one of these to be. So we use the OR term. And then in 6, 7, 8, and 9, or 7, 8, and 9, these are the terms that were used for the surrogate specific search term. In 10, this was our correlation term. And then we would also add in a disease indication term for each search. This is the second strategy that we supplemented our initial one with. And you can see that it's similar, except that the surrogate language is no longer in. We see our disease specific search term. And then instead of the surrogate language, we have here FEV, or forced expiratory volume, as our surrogate specific search term. So we searched across 37 chronic disease indications, and listed here are the ones that we searched for. So there's a wide variety. We see Alzheimer's, asthma, infectious diseases, type 2 diabetes. So we really wanted to look at all of these. And just also want to note that these are endpoints that are used both in accelerated approval and also in traditional approval. So it's a mix. So just talking about some preliminary results, the screening is still ongoing. But what we see is a wide variability. So there are some disease indication searches where there are a ton of results that we got. For example, if you look at hypertension, there were over 5,000. And what that means is there's typically also a large number of papers that pass our initial title abstract screen. And there's likely that there will be an article that matches our inclusion criteria. We also see kind of on the other end, there are some indications where there were very few search results, maybe less than 10. And none that passed our first title abstract screening, and probably unlikely that there's going to be a paper that will match our inclusion criteria. So these are the other indications. So what we really see is kind of where there's a lot of search results, some where there's none, and then kind of all in between. Yeah, and so I'm just going to go through some examples of some of the papers that have passed our full text screen and that we want to include in our analysis. So starting here for type 2 diabetes, with the surrogate endpoint of serum hemoglobin A1C. These are largely meta-analyses where they looked at several randomized control trials. And what we found for type 2 diabetes is that there was largely no association found between HbA1c and the clinical endpoint of mortality or cardiovascular events. However, we did see it really is going to be kind of a mixed picture. So for example, for osteoporosis, on the other hand, we did find 2 papers after our screening that did find significant associations between the surrogate endpoint of bone mineral density and the risk of fracture. As another example, if we look at gout and the surrogate endpoint serum urate, there was a meta-analysis for it, and there was no association between serum urate and the risk of gout flares. So really what I think we'll see for each of these indications is 3 possibilities. There's one possibility where there's no meta-analysis, no systematic reviews identified through our search. There's another possibility where there is a meta-analysis identified, but no association was found. And the third is that we find a meta-analysis and the association is found to be strong. So those are the 3 possibilities. And for each of these disease indications, we want to go through these searches and parse out what the evidence is for each of them. So that's kind of where our next steps are to finalize our list of included systematic reviews and meta-analyses, extract the appropriate data from each manuscript, which will include the number of studies included, as well as what the actual interpretation of the association is, and then synthesize all those results to really update that surrogate endpoint table with another column that lists what is the actual evidence for this surrogate that has been used. Some of the challenges that we face are, we designed our search to be as broad as possible with the hope that we're really not going to miss anything. However, we recognize that this is still not going to catch every single paper that might be included. So to kind of mitigate that, we did add in a second search, and we're also going to do some citation chaining where we look at some of the papers that are included in our search. And if there are citations within those that might be included, we want to loop those in as well. Another limitation or challenge is that there are some cases where, although there might not be systematic review or meta-analysis, it could be possible that there's maybe one or several individual clinical trials that support the use of those surrogates. And so those are things that would not be captured in our research because we're looking specifically at systematic reviews. And we also limit our analysis just to chronic conditions. And so there are other indications, other surrogates that we won't be looking at. So thanks for your time. If you have any questions or comments, please feel free to email me. It's just my first last name at yale.edu.
Fantastic. I can't believe your advisor forced you to do 37 systematic reviews. It's very impressive work. 37 systematic reviews. He's like, no comment.
It's not that I forced him. It's that I got another colleague involved who made it far more complicated.
Thank you so much.
Tim's doing an amazing job.
Absolutely. And our final panelist is Mr. Brian Gilbert.
Hi, everyone. Thanks for being here. I just finished a PhD in biostatistics at Johns Hopkins University, and I'm starting a postdoctoral fellowship at NYU Langone this summer. So my work is mostly about causal inference and spatial statistics. So this presentation is going to be a little bit different because it's more relevant towards environmental regulation than clinical practice, but hopefully it'll still be interesting to at least some of you. So I'll share my screen now. Whoops. Okay, so we're going to be talking about an issue known as spatial confounding, which I'll explain what it is later. But just to give a sort of motivating example, we have, sorry, one second, I just want to check the time so that I know when we're, okay, thanks. We had a data set of births in the state of California from mostly the year 2014. And for each person in the study, we had a bunch of socioeconomic and medical covariates or potential confounding variables that we were able to control for. We also measured everyone's maternal residence at the time that they gave birth. And the study question of interest was the effect of fine particulate matter, so small air particles, the effect of exposure to fine particulate matter, or PM 2.5, which just refers to the size of the particles, on birth weight. So as many of you probably know, low birth weight is usually considered an adverse health outcome and can be a warning sign of more serious problems. And there's been some research that has linked a quantity known as green space to both birth weight and air pollution. And I'll explain what green space is, but because of this, we think that green space is irrelevant, but another potentially relevant confounding variable. And green space just, it sort of comes mostly from the sociology literature, but has been adapted more recently in public health and medicine, which is just the amount of green vegetation in a given area. So it would be high in like a forest or a park, and low in sort of the built urban environment or in a desert. And so the question is, if green space were an unmeasured variable, would it be possible to use spatial information itself in order to control for it as a confounding variable? Now, as it turns out, we do have available data on green space, which is measured by a quantity called NDVI, which is just a measure of the spectrum of light coming off a given area, and this is from, I think, remote satellite imagery. So we have a picture of both the exposure to fine particulate matter, which is high in around the cities of Los Angeles and San Francisco, and also in the San Joaquin Valley, where a lot of agricultural production takes place. And so there's clear spatial patterns to each of these variables, and that brings in some specialized kinds of techniques for fitting the data. So there's been some interest in recent years on the question of spatial confounding, which is what we're talking about when we talk about a unmeasured confounding variable that has a spatial structure. Spatial structure just meaning that observations that are closer in space are also more likely to be similar to each other. And there are a few different kinds of problems that you can encounter when trying to use spatial variables in a causal analysis. I'm not going to get into it in detail, but the one that we're going to be focusing on today is omitted variable bias. Omitted variable bias is just what you're most likely familiar with the idea of leaving out an unmeasured confounding variable, the idea that correlation doesn't imply causation. You want to control for all possible confounding variables. But there's some confusion in the literature because there are other things that can go wrong when fitting spatial models, because you have to sometimes use penalized estimation, which favors less complicated spatial functions, which, if you're familiar with the sort of bias variance trade-off in statistics, this can induce bias in the estimation in order to control variance. And there's a word that was coined called concurvity, which is supposed to be similar to collinearity, which is the idea that if you're trying to control for a function, if you're trying to control for a exposure that is itself a smooth spatial function, and you include a smooth spatial function in the regression, those two things are kind of entangled and you can't really estimate it. But we're focused on the idea of omitted variable bias. So that's when there's an unmeasured spatially variant confounder that we're going to call U. And the goal is to leverage spatial information to capture variability in the confounder and mitigate this omitted variable bias. And this is probably what people have intuitively in mind when they fit spatial models. But there's been sort of a disconnect with the causal inference literature where this procedure hasn't really been sort of explored from a causal inference perspective or justified scientifically. And what basically we need in order for this to be possible is for U, the unmeasured confounder, to be a nice function of spatial location. If it's if it doesn't have a strong spatial structure, or if it's a spatial function plus random noise, then that's not going to work. And for the reason of concurvity that I mentioned a little while ago, the exposure itself, your independent variable, has to have non-spatial variation. So the unmeasured confounder should be a smooth function of space, and the exposure should be a non-smooth function of space. And you also want to ensure that conditioning on spatial location doesn't induce confounding. This could be an issue if participants in the study are sort of moving around during the study, but if you think of the spatial location as sort of causally precedent to all the other variables, then this shouldn't be too much of a problem. There are also there are a couple of techniques from the causal inference literature that I'm going to go through briefly that we also introduce here, because in spatial statistics for these sort of environmental studies, often they're fitting complicated functions of space to control for confounding, but they're still, at the end of the day, fitting linear models in the exposure, which is problematic if the effect of the exposure is not actually linear, which includes being homogeneous. So what we're interested in is the effect of a shift of everybody's exposure value. Instead of saying that the true model is a linear model, we can still think about just incrementing everybody's exposure value and assessing how much that would change the outcome, even if that change is not homogeneous or not linear. So this is sort of a generalization of that estimand. And this is pretty relevant for policy because it's the effect of an incremental change. And you're able to summarize a complex process down to one scalar quantity, even though you're not assuming a linear model. And also from the causal inference literature, there's been interest in fitting. You might know that if you're familiar with sort of propensity score models, it's actually possible to assess the effect of an exposure by fitting a model for the by fitting a model for the exposure itself, or as you may be familiar with from just regression analysis, you can fit a model for the outcome. And it turns out that if you do both of them, they can be combined in a way that improves the robustness and efficiency of the estimators. And we're able to show that these asymptotic results apply under certain kinds of spatial dependence. And just to give the results of the analysis, not going through every number, but what we found is that to sort of experiment with this way of controlling for confounding, we could fit a model where we adjust for the green space variable directly or adjust indirectly for it using spatial location. And we find that whether we do either of those things, we get a qualitatively similar change in the effect estimate, which is as expected. And the effect estimate that we get is within the range of previous meta-analyses, and it's about a minus, the units here might be a little bit confusing, but it's a minus 0.15 gram change in birth weight for a 0.1 microgram per meter cube increase in PM2.5. And just to show a little bit something about like the marginal relationship between PM2.5 and birth weight, it looks like this, where the air pollution exposure is on the x-axis and the birth weight outcome is on the y-axis in grams. And it looks kind of like a blob, which is somewhat common in environmental studies, where there's not, it's not going to be like a smoking gun, very strong relationship, but we have a very large sample size, so we can detect small changes. And you have to remember that these are exposures that affect millions and millions of people in the same way. So even if the effect on a given person is small, these are some important policy questions. As you can see, there is some, you could call it heterogeneity or nonlinearity in the effect, as there seems to be a sort of threshold effect where it doesn't necessarily matter what your exposure is at small levels, but then at a certain threshold, there is a negative relationship. So just to conclude, spatial confounding, as opposed to non-spatial confounding, where there's just some unmeasured confounder and you don't know anything about it, is actually a good thing because you can use spatial information to control for the confounding, as long as a confounder is smooth in space and the exposure is non-smooth in space. And it's important to use flexible models to allow for heterogeneity and nonlinearity. In the future, we're also interested in sensitivity analysis because, for example, the idea that the confounder has to be a smooth function of space is pretty restrictive. There should probably be ways to mitigate bias, even if it's some spatial variable that also has some non-spatial component. There could be interference between units in a spatial pattern, which this spatial information will also obviously be helpful for. And then we're also interested in mediation analysis, where I was talking about controlling for the location. You have to ensure it doesn't induce confounding. Another possible issue would be if the unmeasured variable, if there are unmeasured mediator variables, you may or may not want to adjust for them. And if they're spatially varying, then the spatial function that you fit will probably pick up those mediating variables as well. So just to acknowledge who I've been working with, I was advised in my PhD research by Betsy Ogburn and Abhiraf Datta at Johns Hopkins, and the birth weight data came from Joan Casey of the University of Washington. And my references, if anybody's interested in them. And so thank you all for listening.
Fantastic presentation. Thank you so much. Really, and I'm sorry I missteered you, but you're now doctor. Congratulations.
Unsure whether that's official, but I'll take it. Thank you.
Okay. Close enough, right? You've got a job. You have to finish now. Fantastic. Any questions? If not, I can certainly start, but.
Molly, I can ask you a question. It was really, all presentations were great. Jason, I don't know if you're still on or if you're able to answer questions. The work that you did is really very interesting. And I'm curious, as you were putting together your data set, particularly as it relates to inequities, the implications of the having to have had your hemoglobin A1C measured within a certain period of time, I think it was 7 days before or after, in terms of starting the medication. And just given the burden of getting the lab test drawn and then getting the prescription, might that actually have exacerbated, at least biased the sample against pushing people out who are more socially vulnerable, people who had to get the lab test and then maybe come back 2 weeks later in order to get the result because of other problems, and like they couldn't just hang around the clinic or something like that. I was just thinking out loud. And I was also curious, though, just as a tag on, why so few people were from the UCSF system. I feel like that was like a very tiny sliver of your circle. And so I didn't know why that might be.
Yes, so for the second question, there's so few people from the UCSF system because UCSF is usually patients come there for secondary care and not necessarily primary care. So because of that, we don't see as many new type 2 diabetes users at UCSF. And then for the first question, yes, so we do believe that could that's one of the potential limitations of our study, is that it does exclude a lot of patients when we take patients who do have their lab measurements within the plus or minus 7 day window. But what we also notice is that if we do not take patients within that plus or minus 7 day window, we notice that there are actually large gaps between when patients get their hemoglobin A1C and when they actually get prescribed their therapies, which could also lead to more bias in our data set as well. So we just decided to just plus 7 minus 7 and kind of just take that data from there.
I have a question for Dr. Keller and maybe Dr. Zhu as well. So I'm very curious. So both of you are looking at kind of like the next stage in pharmacogenomics, which is figuring out like how people want to use it. I'm really curious what you're finding about how many people are expressing concerns about the relevance or the interest or the evidence behind the pharmacogenomics as they're talking about it, the testing.
As in like the result that's uncovered from the test, is it?
As in, like, how much evidence is there that like the results from the pharmacogenomics test actually changes the outcome for patients, either through like having a different drug or like actually having different results because of the test?
And Dan, if I could just jump in some ways that it actually sort of links to Mariam's first presentation on trust, like an AI/ML, like how was it validated, and like where did the results for the test come from? Do patients understand them? Do they trust that this is actually should be guiding decision making? I was curious about that too, actually.
From the research I've done today, anyway, that is a attribute that some studies have looked at. So whether a patient's opinion can be used then to do they get an input and as to like the direction of their treatment after this comes up. So it does seem to be important to patients. They do want that opinion, but then at the same time, I suppose it is that risk of how much does the physician have to, healthcare provider, listen to a patient? Because I'm someone, like, obviously I'm not clinically trained, I'm a health economist, but I'm a hypochondriac. So if I do see stuff like that, I will be thinking, oh no, I better ask for this type of treatment. But at the same time, I don't know. So it is that balance of, yeah, patients definitely do want to have an input, but there has to be a guidance of a healthcare provider saying, okay, to a degree I can incorporate your preference, but this is my medical opinion.
And from my project is looking at physicians' perspectives. And by far, when we are reviewing all those papers, and we didn't find any specific paper talking about like the physician's trust to those pharmacogenomics knowledge. But basically, they have those relevant questions like, where do you get those knowledge from? And what do you think will be a good knowledge source for you? And the physician think about the guidelines and the journal papers and FDA tables, or those are like, they just trust them. But there's another thing I want to like, I highlighted in my presentation is physicians do think those knowledge kind of a confirmation of their clinical judgment. And they do have concern that if you trust the genetic information too much to overwhelmingly like interfere with your clinical decision making, it might misleading the whole practice because genetic information is only the starting point of the disease. There's epigenetics and there's a phenotype and there's a genotype versus a phenotype. There's a lot of things going on between these. So I think physicians generally, they trust the guideline and trust the formal results. So they don't really go deep to the mechanism of everything. So.
Thanks. Any other questions? We have about a minute.
Hello, I just had a question for Sam, if he's still on. Hey, Sam, thanks for your presentation. I thought it was incredibly interesting on surrogate endpoints. You may have mentioned this, I may have missed it, I apologize, but could you clarify what the criteria were for looking at the clinical outcomes that you chose, in particular, because for any given surrogate, you could look at a number of different clinical outcomes and there may be stronger associations with some compared to others. In particular, I noticed that you showed the association between hemoglobin A1C and mortality. There was no association there, but that's a pretty high bar for any kind of biomarker to meet all-cause mortality. Whereas there could be other outcomes where like cardiovascular death or microvascular complications where you would find a signal and those would still be, you know, clinically significant. So just those things. Could you talk about how you chose the outcome and have you thought about looking at different outcomes for individual surrogates depending on what they are?
Sure. Yeah. Thanks for your question. I think this was part of what we had to figure out, given that in the FDA's table, they list a surrogate, list disease, but they don't list like a particular clinical outcome for that particular disease. For some disease indications, it's like more obvious than others. For example, like in osteoporosis or gout, it seems like pretty clear that the outcome of interest is usually fracture risk or gout flares. But I think you're absolutely right that in other cases like type 2 diabetes, you can see variability. It could be mortality or it could be other events. I think our approach for that has been to make a decision if we think it is really clinically meaningful in that, you know, is it having an impact on patients' quality of life, their symptoms, their survival? And I think what we're going to do is we're just going to report all of that. So for example, for diabetes, we want to include all of those associations and not really make a decision one way or the other about whether like a clinical outcome is more important than the other, but kind of report and include all of them. But it's a very good question and something that we have to kind of parse through and work through as the project goes on is exactly which outcome is the most relevant. But I think you're absolutely right that you could see differences in the associations depending on which outcome you're looking at. So it's just something that we'll have to report and make sure that we're describing well.
Great, thanks so much.
Thank you to all 5 panelists. These were just fantastic presentations. I really enjoyed them. And I think next we have got a break and we are coming back at 5 minutes to the hour. So see you all soon.
Thanks, everybody. All right. Welcome back, everybody. Give people another second or so. Although I'm happy to see Tiana. First on, ready to go. So just as a reminder, as everyone's rejoining, we will go through and have each scholar do their presentation, about 10 minutes, and then we'll have 10 minutes of time at the end for questions. Just keep in mind, introduce yourself, where you're from, what you're doing. Part of the goal of this CERSI Scholars seminar, and to have folks across all the sites, is to kind of build networks and create connections and identify ways that people might think about working together. We made a good connection last time between the personal genomic testing and Miriam's AI ML work, and who trusts what. So, who knows what will come down the way. So I think we can get going, and Tiana, I can hand it over to you to start your presentation.
Yep, thank you so much. So I am a medical student at Yale, and my project is on the concordance of clinical trial evidence supporting approvals for a class of drugs, the oral anticoagulant drugs, across regulatory agencies around the world. So the big picture question that motivated us was this: are different agencies around the world relying on the same evidence when they're approving applications for the same drugs for when they're new and novel? And usually we would assume yes, but there's a little bit of nuance to it, actually, and I'll go over some of the background to explain that. So first of all, there's a question of what submissions drug sponsor companies are required to include in, like, what data is required in their submission documents. And there's the work of the International Council for Harmonization, which is a council with many member countries, and they meet to discuss what they would recommend for regulatory agencies around the world. So their guidelines say clinical data pertinent to the efficacy of a product and relevant data should be submitted to those agencies when sponsors are considering submissions. And something very similar in terms of the phrasing of the guidelines is used by the European Union's European Medicines Agency, and also by the Australian Therapeutic Goods Administration, the TGA. Both of those agencies say pertinent studies should be considered in the submissions. But the FDA actually says something slightly different in terms of the words that are used. So the FDA says controlled clinical studies and pertinent data should be included in the submissions. And Health Canada says something different still. Health Canada says all pivotal trials necessary should be included. And the literature on how different agencies around the world approach drug approvals is quite rich. So there's a lot of comparisons on application outcomes for the same drugs, and also whether the same drugs receive this approvals for the same indications, and also what types of approval processes the same drugs might undergo. The time to approval varies around the world, and also what constitutes a pivotal trial has also been compared. But more specific to our motivating question, there's only 2 studies, to the best of our knowledge, that directly compared the clinical data supporting specific drug approvals. So one study focused on ADHD medications, specifically extended release methylphenidate, and they chose 4 different countries to study. So they chose Australia, Europe, not a country, a continent, the US and Japan. And the clinical trials that were included in the approval documents for the same drugs varied across these different agencies. Another study indirectly compared the data that was submitted to different agencies and different agencies' interpretations of the data in a bigger study that was comparing the different indications that were approved around the world. And so they found that different interpretations of the same data explained the differences in terms of what indications are approved, but also in about 38% of the cases, there was different data submitted for the 100 or so drugs that they looked at. So for our study, we decided to focus on oral anticoagulants partly because it is similar enough, but also different enough from the methylphenidate study that I just mentioned. So oral anticoagulants are also small molecule drugs, but it is a different therapeutic area, so cardiovascular as opposed to a stimulant therapeutic area. And the indication of interest that we wanted to focus on was stroke prevention in atrial fibrillation. And these are the 4 drugs that we looked at. So this was a cross-sectional study, and we focused on the earliest documents that were available, indicating that an agency approved the stroke prevention indication for those drugs. And we used the following 4 data sources. And for the European EMA, the FDA, and the Therapeutic Goods Administration, historic documents were available, but Health Canada did not publish their historic documents dating to the earliest approval for this indication. And so we corresponded with them to obtain those documents. In terms of our data extraction, we wanted to focus on efficacy, and the 6 or so different major areas of efficacy we looked at were evidence of what specific phase 3 trials were included, and then the sample size of the phase 3 trials that supported the primary efficacy endpoint analyses. And we also looked at the specific numerical results for each of those endpoints, and also the agency's interpretations of those results. And separate from the phase 3 trial evidence, we also looked at the phase 2 trials that were summarized in the documents, and then we summarized that information in terms of a count, just a sum of how many phase 2 trials were mentioned in the approval documents. In terms of exclusions, we excluded phase 3 trials for indications other than atrial fibrillation and stroke prevention. And we excluded phase 2 trials that were not relevant to efficacy or safety. So some phase 2 trials that only focused on pharmacodynamics or pharmacokinetics. And we excluded studies that were mentioned but not summarized in the main text of the regulatory document, which relies on the assumption that perhaps those studies were not truly being considered as relevant or pertinent evidence. And Miriam and I performed and extracted the data, and then we resolved any disagreements among the whole team. And so here are some of the results. To explain this figure, which might be unfamiliar to some people, so every line indicates a pairwise comparison between 2 agencies. And these are the different types of lines, which the legend shows here. And in terms of concordance and discordance, that's indicated by the color of the lines. So for apixaban, we found that there was concordance for everything except for the number of phase 2 trials across agencies. And every pair of agencies had different numbers of phase 2 trials in their approval document for apixaban. And something very similar was the case for dabigatran, except for there was concordance between the FDA and the Australian TGA for the number of phase 2 trials. For edoxaban, TGA is missing because, to the best of our knowledge, the sponsor company did not submit for approval in Australia. But for the other agencies, the only discordance was in the number of phase 2 trials. And rivaroxaban was somewhat different from the other 3 drugs I just went through. So for rivaroxaban, not only were the number of phase 2 trials discordant across every pair, but there was also discordance in Health Canada's description of the phase 3 trials, sample sizes, and the specific phase 3 trials that Health Canada included in their document. And that was the major discordance for rivaroxaban. To summarize, we found very high concordance actually for phase 3 trials and sample sizes, except for in the case of rivaroxaban, and that was mostly explained by Health Canada. We also found high concordance of phase 3 trials, primary efficacy endpoint, numerical results, and agencies' interpretation for all 4 of the drugs we looked at across all agencies actually. And the one thing that was interesting, extra interesting for me, was the low concordance of the number of phase 2 trials across all the drugs we looked at. And so some of the bigger implications, could it be that the ICH guidelines toward harmonizing technical requirements for regulatory agencies had a positive effect in terms of how different agencies look at pivotal trials or phase 3 trials. I think to some degree, our findings do imply that. But there's also the case of the discordant phase 2 trials. And some of the reasons could be due to ambiguity in the language that different agencies use for their submission requirements. So some agencies, most agencies do not unequivocally request applicants submit every single trial that was conducted. And there's also the possibility that trials were submitted by sponsors, but the approval documents that we used did not summarize those trials because perhaps agencies consider them not pertinent or not relevant. And so once again, the big takeaway is that phase 2 trials were discordant across the agency's approvals for the 4 agencies we looked at and the 4 drugs that we looked at. So this big limitation is that our motivating question could actually be divided into maybe 2 separate questions. So what data is actually submitted to agencies and what is actually considered pertinent and relevant, and comparing those 2 different aspects across agencies. And our study design is more suited to answer the latter question and not the former. And we also did not directly correspond with agencies, especially Health Canada, to clarify their discordance with the other agencies. And this was only one drug class and it wouldn't apply to other therapeutic areas. Potentially not. There's also that we extracted the phase 2 studies across both efficacy and safety summary sections of the different approval documents, whereas the phase 3 study outcomes that we looked at were extracted mostly from the efficacy sections. And if we were to extend the study, we would look at safety and compare that across agencies. So just want to acknowledge my collaborators and the funding opportunity. Thanks.
Terrific. Great job and a nice project. Our next presenter is Angela Yu.
Hi, everyone. Just getting my PowerPoint set up. Can everyone see okay?
Yeah, looks great.
Perfect.
All right. Awesome. So good afternoon, distinguished speakers and CERSI scholars and attendees. Thank you so much for joining in on today's Yale-Mayo Clinic CERSI Scholar Research Seminar. My name is Angela, and today I will be presenting on regulatory intelligence as a business partner, a topic I've been interested in as a master's student at Johns Hopkins University and as a regulatory professional in the industry. So just a little background on myself. I've been in the biotech industry for 12 years, and out of the 12, 7 has been in R&D research and 5 has been in regulatory. While pursuing my master's in regulatory science, I'm currently working full-time at a medical device and diagnostic company in San Diego called Werfen, and I do a lot of product registrations, MDR reporting to the FDA, complaint handling, and just making sure that we are compliant with all FDA regulations and global regulations. Sadly, this is my final semesters at Johns Hopkins, but I really couldn't think of a better way to conclude my educational studies, and being here with you all today has really, given me motivation to continue my education, seeing all the PhDs and doctors and med students here. It's not intimidating at all. All right, in today's presentation, I will be discussing what regulatory intelligence is, RI in short, and why it's important for both institutions and industries. Then I will talk about some challenges we face as RI professionals, where we can find helpful resources to help ease these challenges in our profession, and end the presentation with the future of regulatory intelligence as part of the overall organization. So what is regulatory intelligence? To quote from the Regulatory Affairs Professional Society, regulatory intelligence is generally the monitoring, gathering, and analyzing of publicly available and experience-based regulatory information needed to develop a strategy for time and cost-saving product development. So the really key words that you want to focus on today is monitoring, gathering, and analyzing. The monitoring, gathering, and analyzing of regulatory information from these areas as shown on the slide, such as user data, research, and just the news you see on TV or online is a big part of a regulatory professional's role. But to integrate these information to something that is understandable and tangible to the company or to your research or to organizations, it is truly the key to unlocking a regulatory intelligence professional's value. So to provide a few examples from today's CERSI seminar, taking gene and cell therapy, a technology that is constantly innovating and evolving. The agency currently demonstrates their desire to explore surrogate endpoints, as we've heard from Sam's presentation today. And they've been really looking into this, especially in accelerated approvals. Taking the Duchenne muscular dystrophy drug Sarepta, despite promising long-term outcomes, the regulatory intelligence here is knowing that even with an approval of any kind, it might still not be the end game for this particular drug. Drugs lacking clinical response data will probably face reimbursement issues later on. And this is a situation where it becomes a barrier to deliver gene and cell therapy to patients. And another example from today's presentation will be breakthrough therapy designations. Companies also need to keep in mind that reimbursement is not standardized upon approval. For instance, the Medicare coverage of innovative technology, MCIT in short, was rescinded as of last year, and companies must consider if hospitals and clinics will cover non-Medicare products. The good news is, as of 2023, a new act called Ensuring Patient Access to Critical Breakthrough Products was introduced by lawmakers. This will require Medicare to temporarily cover all medical devices approved by the Breakthrough program. So I'm not saying the regulatory team is an expert in everything from drug development, medical device development to reimbursement. My point here is that using these two examples, the regulatory team in the company can help ensure different aspects are considered early on during the development phase and prevent any delays that might be downstream to prevent or create barriers of products reaching end users. So this leads us to the discussion of some of the challenges that regulatory professionals face currently. In a 2019 survey, regulatory professionals were asked which areas of regulatory they worked on, and the majority of respondents, 71%, said the pre-approval and therapeutic space was the most common area of focus. We can also see the other extent of areas that regulatories are responsible for. Keep in mind that this survey only covers the pharmaceutical area and not the medical device or in vitro diagnostic sector. So the takeaway message of this slide is that with increased complexity of technology, with a broadening of innovations, there's definitely going to be demand in regulatory professionals and also a demand in the knowledge and skills that regulatory professionals pick up during their career. Furthermore, in Tiana's presentation, we know that there are a lot of geographic locations to cover. In just the US alone, apart from the FDA, there are over 220 active federal agencies, and it includes the DEA, the USDA, and the CMS, and etc. Internationally, there are more than 50 drug and medical device authorities, and in some countries, regulatory functions are performed by the Ministry of Health because they don't have their own regulatory agencies like the FDA. So this creates an issue where the information online can be limited or ambiguous, or there can just be a large volume of information for us to handle. And because of all this, it adds an additional complexity and expectation and requirements for the regulatory team. I also wanted to note in our company currently, we are approved in 78 of these countries, and all 78 of these countries are information that we have to manually keep track and manage. Another look at regulatory job postings on LinkedIn, it further demonstrates the surprising amount of diversity regarding the regulatory professional's responsibility. For instance, for the same job titles at 2 different companies, we see that the gathering of intelligence is not a common aspect of the job description. Furthermore, regulatory intelligence is often conducted informally and not always documented in sufficient numbers of position descriptions. With that said, in order to help collate information across all these different markets and all these different international requirements, there are ways that we can find regulatory intelligent resources to help mitigate the complexity of our issues. It can be staying educated on FDA's current thought processes through experience, or inviting the FDA to present at professional meetings like today. In addition, ongoing education and training, networking through professional associations like RAPS or AdvaMed, and staying on top of current news and trends that we see on the news or read online. These are all ways that can help identify and find information that not only regulators, but R&Ds and researchers can use to integrate in-house. Lastly, it wouldn't be fair for me to end my presentation without mentioning artificial intelligence. So artificial intelligence is very fast moving, but it has to be seen as a tool for regulatory intelligence, which improves efficiency and transparency. So currently through our CERSI program, the FDA has already been advancing the understanding and usage of AI to improve regulatory review and post-market surveillance, as seen from a lot of the earlier presentations today. When compared to traditional regulatory intelligence, artificial intelligence offers multiple benefits. As shown in this image here, it includes automated monitoring of regulatory information, reduction in time for searching all those data, and its AI will also help RAs retrieve regulatory information much quicker in a much more manageable fashion. In addition, a thought I had that would be promising in the future would be some sort of chat box to help us do our pre-submissions. I did some research online about current chat boxes, and it includes Dialogflow, Microsoft Bot, or Raza open source. So how can these chat boxes be useful for our profession? Well, we can potentially use it as for regulatory-driven pre-submission questions, such as interacting with the chat box to ask, what is a typical sample size for a clinical trial for, let's say, infusion pump? What are the demographics required for the clinical trial for an infusion pump? What are some complications that have been reported for the device? What are some safety communications released by the FDA in 2023 regarding these products? And last but not least, are there any current infusion pumps or X product currently marketed as safe and effective? And are we able to take like an FDA 510K route? Of course, before we dive into and start using chat boxes, the effectiveness of this platform needs to be validated and monitored. So to summarize my presentation today, regulatory intelligence professionals play a pivotal role in the organization with respect to compliance, optimize approvals, and launching safe and effective products on the market. To be an effective RI professional, we must also become an educator for cross-functional training. And what does that mean? It means we need to be generous with information sharing, like today in our seminar. We have to embody technical and people skills. In addition, altruism is also very important. From my experience, being open to sharing information, but not taking it personally when different departments doesn't accept the information is also a good trait to have. Regulatory intelligence is also not only critical for companies, but a global benefit. And that means that with continuous scientific innovation and changes in regulations, not just in the US, but globally, an effective contributor will help be able to better manage FDA expectations and other agency requirements. Next one, using AI as a tool for improvement. It has a lot of potential to bridge the gap between end users who seek the information like companies, and also bridge the gap between R&D and researchers that need the information to conduct their clinical trials or investigation. And finally, regulatory intelligent participation at the highest levels within companies should be conducted as a business partner. This ensures quality is built in the whole business ecosystem. So this concludes my presentation on this topic. Thank you everyone for making time on Cinco de Mayo to tune in, and this has been an amazing experience. If you have any questions, feel free to ask during the 10-minute Q&A or through this email I have here.
Thanks, Angela. That was really great, and it's great to see kind of how people may use the kind of information that we're generating at the academic side. So that's terrific. Magdi El-Salab, yeah, great.
So,
and please remember to introduce yourself and let everyone know.
Yeah, sure. I'm just waiting. Yeah, for Angela. Yeah.
Yep. There you go.
Can you see my screen? Yeah, perfect. So, hi everyone. My name is Magdi El-Salab. I am a research fellow at the Harvard MIT Center for Regulatory Science, and I'm a medical doctor by training, and I got my PhD in regenerative therapies, including gene therapy and cell therapy, and I have been focusing on regulatory science questions since my PhD. So it's my pleasure today to introduce one of the projects that I am working on, and it's looking at the probability of success of gene and cell therapy development programs. Just very quickly, what is the current FDA definition of gene therapy and cell therapy? The FDA defined gene therapy as therapeutic agents that mediate their effect through transcription or translation of a transferred genetic material, or by altering the host genetic sequence. While cell therapy is defined as any autologous, allogeneic, or xenogenic living cells which have been manipulated ex vivo, and the manipulation can be propagation, expansion, selection, or pharmacological treatment. One of the examples is the mesenchymal stromal cells, while one of the examples for gene therapy, of course, is chimeric antigen receptor T cells or in vivo genetic modification using viral vectors. And the other thing is what is very common, or what is common among these new class of therapeutics, and what makes them different from other classes. They are considered as living drugs, so they are not easily characterized similar to small molecules or simple biologics such as enzymes or antibodies, and they are not usually consisting of one simple cellular population that you can characterize similar, for example, to small molecules when you can have the chemical structure for this molecule. And some of the examples, as I mentioned here, are CAR T cells and viral vectors, which can be lentivirus or adeno-associated virus. So how these products are regulated in the United States? There are currently two routes for regulation. One is the 351 pathway, and the one is called the 361 pathway. The 351 pathway is the approval pathway that require the FDA evaluation of the pre-marketing evidence before release to the market, while the 361 pathway does not require the FDA approval and is usually reserved for products that are considered minimally manipulated. While if you have, according to the definition that I presented earlier, more than minimal manipulation, these products needs to go through the FDA and need to comply with a good manufacturing practice regulation. So how does the gene and cell therapy approval landscape look like? We have currently 19 gene therapy and cell therapy that has been approved in the market. And we can see on the bottom left that we have been seeing a constant increase in the number, especially for the last 2 years. And the FDA is projecting to approve between 10 to 20 gene and cell therapy by 2025. The common thing about these products that most of them are the product of the academia. And usually the story, and I have here 3 stories for gene and cell therapies, start in the academic institutes, and then a small a spin-off or a small and medium-sized enterprise come to the play and develop the product further. And then the big pharma usually come in the end and acquire those companies and go for the approval. And this is because this is a high-risk development field that is not well characterized, and there's high risk of failure. The other thing that I think play a big role in the development of gene and cell therapy, that it's the life cycle of the product itself is different, and this uniqueness is across the whole development, from the preclinical to the manufacturing that is being used, to even the target indications, as usually they are orphan drugs that has a small sample size. And of course, when we go to the post-marketing, the need for the reimbursement, which is still not as developed as for other product types. And this was the motivation for us to see how this kind of environment affect the success rates and the probability of success of gene therapy and cell therapy development programs in reaching the market. And if the regulatory environment and the regulatory incentive play any role in changing this probability. And the last thing, how this probability change throughout time. And I'm presenting some of my preliminary data. First of all, what was the data source? The data source that we have, and this is something that is challenging for conducting such kind of studies, because it's hard to track a compound from the early development to the approval, especially in the case of failure, because the products get acquired by different companies. Some of the trials are not registered. And this is why it's usually not an easy task to track the products and to try to accurately estimate the probability of success. So we had an access to a database. It's called ADIS Insight, and we tried to obtain all the drugs and the trials that are labeled as gene therapy and cell therapy. But then, to overcome the limitation of this inherent missingness of data in such databases, there was a new development in this area, where there is a new method of estimating the probability of success, which try to impute any missingness in the database. And as we can see here, usually in such studies, what has been done before is to look at the trials that are in a certain phase, and then look how many of those trials advanced for this compound, advanced to the next phase, and then divide the products in the advanced phase over the product in the original phase. And if you have missingness in your database, you might underestimate the probability. So for example, for the drug development program number 1, there was a missing phase 2 in the database. And this will lead to an estimation of phase 1 to phase 2 probability of success of 2/3. While in reality, because there was a phase 3 in the database, you can assume that there has been a phase 2 that has been conducted along the way, and that was not captured by this database. And this is why you can impute this phase. And in that case, your probability of success from phase 1 to phase 2 will jump to 100%. This method, or this model, was developed by a group in MIT, and it's called the path-by-path approach. And as we can see, it relies on breaking down the clinical trial to by phase and indication. So if a clinical trial targeting multiple indication, and this is especially true for early phases, in phase 1, where safety is tested, for example, in case of chimeric antigen receptor T cell in B cell malignancies, and this can include acute lymphocytic leukemia and non-Hodgkin lymphoma. And then after a signal is detected, this signal is then confirmed in further phases like phase 2 and phase 3. Another important definition in applying our model is the lead indication, which is the indication that progressed the furthest in the development compared to the other indication. And again, this is the imputation strategy that I elaborated before. And another imputation method that we used here is to impute the status of the phase or the status of the clinical trial if it wasn't there. So the model assumed that after the end date, if there is a certain period of time, if this period of time elapses without the appearance of a phase in the next stage, we assume that this trial or this phase was terminated. If this period was not elapsed, then we will consider the phase as still in progress. So after extracting all the drugs that were listed as gene therapy and cell therapy, we manually did our inclusion and exclusion criteria, and we ended up with around 1,300 product that corresponds to 3,369 trials. And we used those products and trials to construct our model, and this led to possible development programs of 4,386 and around 4,440 phase transition. And this is the cumulative increase in the clinical trials over time. And we see that there is an exponential increase throughout the recent years. And we see also that this is when we plot this as year by year number of trials, we see that there was a constant increase in the number of trials. We then looked at the disease area and the trial phases in that we acquired. And we see that most of the gene and cell therapy products or trials target oncology indication, followed by circulatory system diseases, and then endocrine and metabolic diseases. And the other interesting thing that we noticed that the prevalence of phase 1-2 trials in the gene and cell therapy area, which compared to the previous studies that has been published, usually the percentage is much lower than 28%. We then applied the model and we did a subgroup analysis based on the indication, the acquisition of an orphan designation or use of fast track or PRIME scheme in the United States or Europe, and finally the use of biomarker. And the first thing when we apply on all the indications that were tested, the approval rates or the probability of success from phase 1 to the approval seems to be low, which around 4%. And when we look at the lead indication only, it's around 10%. We then looked because of the prevalence of the oncology indications, we looked at the non-oncology versus oncology indications, and as we can see, the oncology indication probability of success is pretty low compared even to other studies that looked at oncology indication probability of success, which is around 3%, 3 to 3.5%. We also observed that the use of orphan designation or fast track or PRIME increased the probability of success to 6%, around 6%. And finally, the use of biomarker throughout the development from for various purposes, either to estimate the safety or the efficacy, increase the probability of success to around also 6%, but also increase the probability of success in the later stages compared to other compared to the original phase 3 to approval success rate. The other thing that we tried to look at is how the probability change over time. And we use a competing risk analysis where the two events of interest is the termination or the approval. And we see that in the first 5 years, the probability of success, the cumulative probability of success is around 1%, while the cumulative probability of termination is around 44%. The cumulative probability increased to 5% at 10 years, and then started to plateau between 15 to 25 years, which increased to 10%, but then plateaued at 11%. So just to summarize my outcomes, we see that we have a lot of modern trial design, which is usually the phase 1-2. And we see also, and I didn't point this to wild data, but the use of the phase 1-2 trials and the prevalence of the phase 1-2 trial in my data set actually inflate the success rates of phase 1-2 compared to the other study that has been published in this area. And then the use of fast track orphan designation and the biomarker improve the probability of success. And as we saw that most of the development programs get terminated in the first 5 years, and the probability of success usually plateau between 10 to 15 years. And with that, I would like to thank you, and I would be happy to take any question. And I, of course, thank the Center for Regulatory Science and MGH, Mass General Hospital, where I also work on the group with other related regulatory science projects. Thank you.
Thanks, Magdi. That was totally fascinating and very cool. Access to data that not everybody has. I've been I'm doing a bad job of keeping us on time, so we're running a little late this session. Niraj, you're next, and then we'll probably only have like 5 minutes for questions for everybody. So just keep that in mind, just so we don't fall too far behind. But Niraj, you're up. Thank you.
All righty, perfect. Thank you. Can everyone see my my slides?
Yep, okay.
Oh, perfect. All righty, so I'll go ahead and get started then. So hi, everyone. My name is Niraj Patel. I'm a second-year medical student at Yale. And my presentation this afternoon is about an investigation into the informational quality of television advertisements for drugs that have received FDA breakthrough designation. And so I don't have any disclosures to report. My research mentors for this project are Dr. Rashmi Ramachandran and Dr. Joe Ross. And we also worked with Ayman Mohammed, who's a medical student over at Mount Sinai. And so I just want to start out by sort of saying a key motivator for our research here was this idea that patient understanding of the risks and benefits of prescription drugs is paramount for sound health decision making. And so two of the many trends that have sort of challenged this. So one is some confusion that's been created as an unintended consequence of the FDA's breakthrough designation program, which I will get into in a second. And the second is the rise in direct-to-consumer pharmaceutical advertising over the past 25 years, which has been controversial in itself. And so, just to start off, what is the FDA breakthrough designation pathway, and what is the unintended consequence that I ominously alluded to? So for background, FDA breakthrough designation was a program that began in 2012, and essentially the FDA awards investigational therapies with breakthrough designation based on preliminary evidence before phase 3 trials are conducted that these drugs may end up being particularly effective. And the preliminary evidence does include use of surrogate endpoints, as Sam and Angela told us all about earlier. And then when drugs are awarded this designation, the FDA will help expedite development and regulatory review to kind of triage these products. And one criticism is that the name can be quite misleading because, as I mentioned before, the designation is awarded before phase 3 trials are done. And so these drugs may end up falling very short of breakthroughs as we might think about them in lay terms. And some empirical research has indicated as such. And two studies that I wanted to quickly highlight. There was a randomized study in 2015 that found that participants exposed to short factual descriptions of a recently approved drug were significantly more likely to rate the drug as very or completely effective if the description stated the FDA called the drug a breakthrough, which is something that does routinely happen in press releases for these sorts of drugs. Participants were also significantly more likely to believe the evidence supporting the drug is strong or extremely strong. And another really quick study I wanted to mention was a 2016 survey of physicians that showed that 52% of respondents incorrectly believed that strong evidence, that is like randomized trials evaluating clinical outcomes, were required to receive FDA breakthrough designation. And so the other trend that I referred to was direct-to-consumer pharmaceutical advertising, and the prevalence of such advertisements, particularly via television, has risen dramatically since 1997 as a result of some regulatory changes. But concerns have arisen regarding their low informational quality, including distorted portrayals of benefit and misleading promotional aspects like off-label promotion. And so the research question that we wanted to get at really tries to evaluate these two trends in the context of one another. So we're trying to study the characteristics of prescription drug TV advertisements for indications that receive breakthrough designation. And so very briefly, a summary of the methods that we used: we were using an online advertising database called AdPharm to extract advertisements that were for breakthrough designation indications. Two coders assessed those ads for a predetermined set of criteria we created concerning informational quality and promotional language. And then as another aspect of this study, we also wanted to get a sense of how transformative these products typically were. And so we used a peer-reviewed educational bulletin called Prescription International, which provides therapeutic value ratings for new products. And so the first finding I wanted to briefly discuss was that these advertisements tended to emphasize, product newness, uniqueness, and the concept of hope. And there are some examples of that listed on this slide. But the one thing I really wanted to emphasize here was that 9% of these advertisements were using the word breakthrough in their promotional language, which we felt was notable in the context of the empirical literature that I previously described regarding how the word breakthrough can be quite misleading to patients and physicians. And the second finding that I wanted to highlight was that we found quite poor informational quality with regards to statistical claims in these TV advertisements. So in particular, advertisements at times would quantify improved outcomes for study participants taking the advertised drug, but would omit the outcomes for comparator groups. The example shown here for that in the first sort of row there was for a diabetes drug for Ziga, which included an A1C reduction of 1.2 points, but omitted that the same trial saw a mean reduction of 1.4 points for metformin. And then, in addition to that, I wanted to briefly mention that when comparator statistics were included, they were often de-emphasized in various ways, and other advertisements failed to describe, for example, whether a risk reduction was relative or absolute, which can have a huge impact on someone's understanding of what the statistic really means. In terms of the Prescription ratings, 12 of the 17 advertised drug indication pairs had been rated by Prescription, and none were rated to be an important or major therapeutic advance. And then only 5 were rated to offer at least some value versus existing alternatives. And so, the thing that I sort of wanted to end on here in terms of policy opportunities and potential areas for investigation on that front, the FDA could consider regulating against the use of words like breakthrough that are known to be misleading, and issue guidance to manufacturers that assertions of breakthrough designation and marketing materials be accompanied by descriptions of what the program actually means. The FDA could prioritize breakthrough designated product advertising in their issuance of warning and untitled letters to manufacturers, as well as pre-distribution advertisement review. Policymakers might consider changing the name of the program to something that might be less likely to mislead, like the FDA Early Potential Program is something that we thought up. I'd love to hear if people have other name ideas as well. And policymakers might also consider transitioning FDA advertising review to a more proactive system in which all advertisements are reviewed prior to their public dissemination, as opposed to the current system in which many of these ads are reviewed retroactively by I think a very like sort of underfunded aspect of the FDA's regulatory scheme. And that was all I had, but thank you so much, everyone.
Great job, Nirj. And I hope that his paper, when it does come out, will be as written, because you'll see how he managed to quote both the Supreme Court and Shakespeare while discussing direct-to-consumer advertising as a breakthrough drug. So
yeah, I forgot. That was the main
open it up. That was the main finding. Curious if people have questions for anyone. I know I actually have one interesting reflection for Magdi, if just to ask if someone else doesn't have a question. But I was so interested, you know, just the rates that you were able to identify, the very high sort of transition from phase 1 to phase 2, which suggests that a lot of these therapies are being identified as kind of not toxic, right? But the just substantial drops from phase 2 to 3 and 3 to approval, which, you know, around the efficacy. And do you have a sense of how that compares to cell therapy, right? Like, because is it that hard to sort of predict what to bring into sort of clinical testing in terms of efficacy?
Yeah, this is a great question. And I think the main point is we assume for phase 1-2 trials to be phase 2 trials. So in that case, you will impute phase 1 trial as success. If you consider a phase 1-2 trial, as a phase 2, then automatically phase 1. And this is why this transition will be a little inflated. And it was surprising for us to see how different the landscape of the trials when you focus on the gene and cell therapy compared to others. The study that was published by Dr. Andrew Liu group reported a much lower phase 1-2 trials, and in turn the transition from phase 1 to 2 success or probability was lower than the one that we're reporting.
Got it. Dan, I saw your hand come up first.
Great, thanks, Ross. There's a question for Niraj. You have a great presentation. I just find that research on direct-to-consumer advertising is really interesting. I'm from Ireland, so there's no direct-to-consumer advertising. And I did my MPH at Yale, and learning about it there just really blew my mind. So just really cool to hear about that. So the use of breakthrough. So in terms of like marketing advertising in general, not just related to pharmaceuticals, you know, companies can obviously inflate their products to obviously sell them. So how can the FDA then regulate against the use of a word like breakthrough, where they're not necessarily saying that it's going to lead to significant improvements in health outcomes, but at the same time, the word does imply significant benefit. So like, how do you think they could directly regulate that? I think it's a really good point.
Yeah, no, thank you so much. And thanks for highlighting that. You know, I think an interesting thing for me is that FDA the sort of pharmaceutical advertising is only widely permitted in the US and New Zealand, which is an important contextual point. I think that, you know, I am certainly not a lawyer in terms of you know, understanding the exact legal mechanisms, but I think that my guess would be that in terms of how advertising for prescription drugs are regulated, there is you know, like statutory law in place that like highlights that that those sorts of ads specifically for prescription drugs should not be misleading, and there are like examples that are like outlined in, I think it's the Prescription Drug Marketing Act of 1987, of like some of the things that might qualify as misleading versus not misleading. An example is like off-label promotion of a drug is something that is, you know, the FDA, to my understanding, explicitly bars, and that they can kind of draw from that statute to kind of derive that regulatory authority. And so I think that from like a legal mechanism, you know, I would guess that would probably be the route, is like making the argument that certain words that are shown to be misleading are in tension with that aspect of the kind of statutory requirements for such advertisements. But I'm sure you would have different interpretations of what qualifies as misleading versus what doesn't qualify as misleading, which I would imagine could be argued in courts.
Great, thanks. I look forward to reading the paper.
Yep, Dr. Chang, last question.
I have a question for Angela, you about the AI in the regulatory intelligence. Does any of the AI chatbots allow you to have the, for example, accurate and exhaustive list of those trials for like a disease or indication specific topic? My purpose is like, say, does it allow us to check whether AI checkbox like missed any trials or mistakenly included any trials that shouldn't have been included?
That's a great question, Dr. Chen. So as of now, the FDA hasn't used chat boxes for regulatory and industries to do submissions. They are all very manual. We have pre-submission meetings with the FDA where we have conference calls. But moving forward, I know the FDA is working on a lot of machine learning to develop these platforms. Chat boxes, I don't know if it's going to be in their pipeline, but they definitely need to source a lot of data in order to incorporate all the data sets that you mentioned earlier. But as for now, there are no AI chat boxes for us to utilize.
Because I think that's very similar to, for example, the mesh terms, right? So like searching through the mesh terms or like systematic review searching. And I also think like have the chatbox AIs integrated some of the analytical functionalities. For example, you may ask it like say, what's the typical sample size for like a specific phase of a trial for a disease or indication, right? So, and can it give us the like minimum, maximum, median? So I wonder if any of the, like say, AIs you played with actually has integrated the like analytical, basic analytical functionalities.
No, it has not yet. And as I mentioned earlier, we don't have a function right now to actually ask a chat box on what is the sample size, how many physicians or nurses must be included in a usability study. But I hope moving forward, when they do develop the platform, they can use information from already approved products and also data set requirements from guidance documents and pull that all together so that when we ask our questions through the chat boxes, it can give us these parameters or specifications that we can follow when doing a clinical trial.
Yeah, because my first point of accurate and exhaustive list of trials sounds to be like very similar to a work of like a medical librarian. Yeah.
I'm sorry to cut it off because we've already gone a bit over time. It sounds like you guys should set up a time to chat.
Definitely, definitely love to chat.
I'm gonna hand the baton back to Ben for the fourth session.
Thanks, Joe. So our fourth and final session has 4 really interesting sounding papers, each with particular sort of clinical and physiological aspects to them to explore. So we'll start with Dana Motabar from UMD.
Hey, okay, let me just share my screen quickly. Okay, can everyone see my screen? Is that good? Yeah.
We can.
Perfect. Okay. Hi, everybody. Thanks so much for making it to the last session. My name is Donna Motabar. I am a graduate student at the University of Maryland, College Park in the Bentley Lab. So today I'm going to be talking about my dissertation work. In this work, we have developed analytical tools that will allow us to analyze the quality attributes of antibody therapeutics. We're hoping this work can improve bioprocess monitoring approaches and thus help pharmaceutical companies make more consistent and reliable processes for their drugs. Okay, so first I just want to start by providing some background on the motivation of our work. So antibody therapeutics, as I'm sure many of you are very familiar, have come to constitute one of the most rapidly growing sectors in the pharmaceutical market. Importantly, throughout the development and manufacturing processes, product quality attributes or CQAs need to be characterized. And this includes something like antibody variants that are produced due to reduction in oxidation reactions or redox reactions. So typically, in order to characterize these attributes, gold standard analytical methods are used. This includes things like mass spec, liquid chromatography, capillary electrophoresis. These methods can provide very detailed characterization of the therapeutic, but they're generally known to be expensive, time intensive, and do require specialized knowledge. So in order to improve bioprocess development and manufacturing, the Process Analytical Technologies, or PAT initiative, was launched by the FDA. PAT are basically a broad set of monitoring techniques that can assess the quality attributes of the therapeutics. So one of the major principles of PAT focuses on the development of in or online monitoring tools that can provide more frequent and automated measurements as compared to your standard at-line techniques. So when I say on or in-line measurements, I mean tools that can be used to take measurements in the process stream, whereas with at-line techniques, you have to go, sample, and then take a measurement away from the process stream. So hopefully, by using these on and inline methods, will enable the creation of more robust processes, and that can lead to lower development times and costs. So these technologies, I just want to say, are not necessarily meant to replace the gold standard ones that I talked about in the last slide. Rather, we're hoping they're going to complement them by providing rapid, cost-effective, and high-throughput approaches that can be used for process monitoring and control. So in this vein, we're hoping electrochemical methods we're thinking that it'll be a good fit for this application. Electrochemical methods allow for rapid, sensitive measurements, as well as connectivity to electronics and control schemes. And that's been shown previously with the use of these electrochemical pH and DO probes in bioreactors. So they've been standard measurement tools for decades. So now I'm just going to focus in more on our specific approach, which is called mediated electrochemical probing, or we'll refer to it sometimes as MEEP. And how it works is analogous to how bats use sonar to hunt for prey. So these bats will transmit a signal that will interact with nearby prey, and then that signal will be reflected back to the bat, where it can receive and process that information about its local environment. So the scheme on the right gives an overview of our approach, which works very similarly to that. So first, we impose electrical inputs that can code these redox mediators, shown in orange and purple, into their reduced or oxidized state. These mediators are purposefully added into the solution, and they serve as electron shuttles, since electrons can't exist in free solution by themselves. So then the mediator will diffuse and interact with biological elements in the local environment through the exchange of electrons. And then the mediator can return to the electrode, where the information about its redox state can be decoded by the electrode to generate an output signal. And then the output signal can be further analyzed by signal processing methods. Okay, so now that I've given an overview of our approach, I want to show now how we could how it's going to be applied to bioprocessing applications. So in bioprocessing, as I mentioned previously, reduction and oxidation events can occur that can lead to reduced and oxidized proteins. So here we're going to be specifically focusing on monoclonal antibodies, or mAbs. So reduction primarily occurs in the development process, during upstream, in the bioreactor and centrifugation, and this can result in a myriad of these low molecular weight reduction fragments shown on the left there. And that will lead to the exposure of the amino acid cysteine, which has this thiol group. That's going to be important for our method. On the other hand, oxidation occurs through all stages of development, including storage, and it primarily will affect the amino acid methionine. And basically, this is important because these alterations can have product quality effects. So this is why they need to be characterized, and this is why the FDA cares about that. So now we want to use our approach to characterize these reduced and oxidized variants. We're going to, again, leverage those cysteine and methionine residues to characterize our mAbs. And I'm just going to jump into our next slide, which will go over our approach for analyzing those cysteine residues that I said occur when the mAb is reduced. So what we're using here are two electrochemical approaches. One is cyclic voltammetry or CV, and the other is chronocoulometry or CC. I'm not sure many of you are familiar with these approaches, so I'm just going to give like a quick overview of how it works. So with CV at the top, a voltage range is scanned and the current response is measured with respect to time. CC is a little bit differently. The voltage is held static and then can be stepped to another static voltage. As I said previously, we're using these things called redox mediators that act as electron shuttles in our approach. And the mediator we're using for this approach is called ferrocene, or FC, as I'll be referring to it. So up top in the pink and at the bottom also is the control responses using ferrocene. You can see for the CV, it creates this duck-shaped response with similar amplitudes in the two peaks. Whereas, and then at the bottom here, you can see it goes downwards and then switches to upwards when you change the voltage. Now I want to show how our approach can interact with the amino acid cysteine, because of course that's our amino acid of interest. So at the top here in blue, you see when cysteine is added to ferrocene, you're getting a dramatic alteration in that duck shape. You're seeing an amplification in that oxidative peak and a reduction in the attenuation in the reductive peak. Same with the CC approach at the bottom. And so I just want to provide a background on why this is occurring, because you may not be very familiar with this approach. So on the top there, you see that there's a reduced mediator shown in light red. We're going to purposefully oxidize it with the electrode. And then a mediator can cycle with the amino acid, causing the regenerated of the reduced ferrocene. And that's going to result in the amplification of the oxidative peak. For the reductive peak on the bottom scheme there, since there will be less oxidized mediator interact with the electrode, we're observing attenuation. So that's why you're really seeing those changes in the peak when cysteine is present. Okay, so now we've shown that our approach works with cysteine. We're going to apply it to the reduced MAB. So up top is our reduction protocol. We're taking NIST MAB that NIST kindly provided us with, and we're just adding a chemical to it that will create all these exposed disulfide bonds. Next, we just quickly wanted to confirm that our MAB was actually reduced. So we use those standard bioprocessing approaches, these at-line approaches that are typically used by drug companies to analyze these drugs. So on the left, we have this gel, and basically the takeaway here is that in lane 2, that's your intact protein that hasn't been reduced. You only see one band because it's not really fragmented at all. And then in lane 3, we analyze the protein that's underwent the fragmentation process. You can see what we would expect, a bunch of these lower molecular weight fragments. And then just on the right, we also ran a free sulfhydryl assay, which just measures the thiol content of the sample. And again, you're seeing what you would expect. When your antibody is reduced at those different concentrations, you get a concentration dependent response where higher protein concentrations have higher free thiol content. So great, now we've established that our protein is reduced, and we've established it using approaches that these pharmaceutical companies use. So now we want to apply our actual approach and see if it can be used effectively to discern between the intact and reduced states. So here we're adding again the ferrocene to our reduced protein and the intact protein. And these are our results. So the pink colors are the intact MAB at different concentrations, and the blue is for the reduced MAB at those corresponding concentrations. Basically, the takeaway here is that both of the CV and CC methods show two important things. One is that our method is able to detect antibody reduction for all the concentrations we measured. And second, we observe a concentration-dependent response. Higher concentrations of antibody are yielding larger current responses, which is likely due to that free thiol content. So now that we have our results, a big advantage of our approach is that we can quantify them using what we call signal metrics. Signal metrics are really important in bioprocessing because they allow for comparative analysis. And I think maybe this analogy is helpful. It's very similar to an EKG, where a plethora of complex information can be extracted from a shape of a curve. And the plots on the left here just show how we're defining the different areas of the curve, and the right table shows the equations and identifiers for the metrics we apply. So what we did is we calculated the metrics, applied a statistical analysis and a logistic regression model to our data. And what we found is that out of those A through G metrics, certain ones work better for our purpose. Overall, you can see up top in that bar graph, we found that metric C is really able to provide good discrimination between the reduced and intact MABs. And then on the bottom plots there, we used a logistic regression model to see if we can use multiple of those metrics cooperatively to increase the discerning power of our results. And we found that for higher concentrations, as shown in the plot on the left, that purple line, you see that you get that 90-degree curve, 90-degree line, which basically is saying that you only need one metric. Metric C is best suited. It has very good predictive value for when your protein is intact or reduced. However, at the lower concentration in the plot on the right, for like our lowest concentration, 0.25 gram per liter, you really need to use multiple metrics in order to discern the results. You see that when we use 3 metrics, C, A, and D, altogether, you're able to get very good discrimination between your 2 protein states. So I think I'm running up on time here, so I'm not going to talk about the oxidative approach. But I just want to say we did use it for oxidized protein as well, and it worked pretty good. So we were happy with that. But basically, we're just trying to say here that we think our approach is a really good method for detection of antibody redox variants. It has a quantitative aspect to it, which is great for bioprocessing settings. And we think that it can be very helpful for drug development. And just want to thank my advisor, Dr. William Bentley, all our collaborators, and our funding sources. Okay.
Awesome. Thank you, Dana.
Thank you.
Amazing presentation. Great, work and great visuals, and apologies for bungling your name when I introduced you.
Oh,
so.
no, you're fine. Dana is always pronounced Donna here, so it's always very common.
Thank you. So next we have Ritika Kurian from Maryland about CYP2B6. Floor is yours. See, there we go.
Yeah, so hold on. Yeah, hi everybody.
Hi there.
So I'm going to start sharing my screen right now. We just had like a fire alarm go off in our building, so I had to like move. So I hope, can you all see my screen?
We can.
Okay, great. So I'm trying to get to full screen. Yeah, so hi everybody. My name is Ritika Kurian. I'm a 5th-year PhD student at University of Maryland, Baltimore in the Department of Pharmaceutical Sciences. So today I'll be talking to you about my project, which is on augmentation of cyclophosphamide-based chemotherapy through CYP2B6 induction. So just to give a brief background about xenobiotic metabolism. So it's basically a process where you have your drug or a foreign substance which undergoes like metabolism in the liver. So you get, like as shown in the figure here, a drug upon entering the liver gets converted through these enzymes. So it's an enzyme mediated process, and you get inactive metabolites which can get eliminated from our system. So the major goal of metabolism is detoxification. But can we look at xenobiotic metabolism from a non-traditional approach to actually get active metabolites? And that is possible because of design of a prodrug. So a prodrug is a biologically inactive compound, which means that prior to metabolism, it is inactive. It has no efficacy of its own. But after it undergoes metabolism, it becomes like active. And there are different prodrugs in the market available. But the prodrug that we are focusing on is cyclophosphamide. It's known for its prodrug activity. It's a chemotherapeutic agent, which is used for different types of leukemias, lymphomas, solid tumors. And so what is the mechanism of action for CPA, cyclophosphamide, which I'll refer to as CPA? So I can see here in this scheme, CPA has to first get converted to 4-hydroxy CPA, which then exists in equilibrium with this aldophosphamide. Now, this aldophosphamide generates this mustard, and the therapeutic efficacy that you get is because of the generation of this mustard. Now, to generate this mustard, this conversion of this CPA to 4-hydroxy CPA is critical. And you have, like I said, it's an enzyme-driven process. So the major enzyme that is involved here is it belongs to a cytochrome P450 family, and it is called CYP2B6. You have minor contributions from CYP3A4 and CYP2C9 as well. So in order to generate more mustard so that we get enhanced therapeutic efficacy, we have to sort of increase the conversion of CPA to 4-hydroxy CPA. And that is possible if we increase the expression of CYP2B6. But in order to do that, we need to know how CYP2B6 is actually regulated. And that brings us to this receptor, which is called constitutive androstenedione receptor, or CAR, because this receptor controls the expression of CYP2B6. And this receptor is a member of a nuclear receptor superfamily. It's mainly expressed in the liver. And here is a scheme of like the direct activation of this receptor. So in the absence of any ligand or small molecule, it's present in the cytoplasm. Once the small molecular ligand binds, these chaperone proteins, they dissociate, and now this receptor translocates to the nucleus. It forms these partners with these other transcription factors. Together, this complex basically is able to recognize response elements which are present in the region upstream of the promoter region of different target genes. And that's how the transcription is initiated for these various target genes. And CAR has demonstrated, at least human CAR has demonstrated preferential induction of CYP2B6 over CYP3A4. So what we are trying to do is use this strategy where we can actually use an adjuvant drug to activate CAR. And because of that activation, we can get increased CYP2B6 expression that can help increase the CPA efficacy. Now, one of the promising candidates that we have is called CITCO. It's a small molecule. It is a prototypical human CAR activator, and in vitro, it has shown some promising results. So as part of this, my project is to basically investigate this small molecule CITCO, its properties such as its pharmacokinetic features, its metabolism, and whether it can be actually used as an adjuvant drug candidate. And the hypothesis that is that CITCO can induce optimal CYP2B6 expression in vivo, especially when given by an oral administration route, which will then help enhance CPA bioactivation and facilitate the CPA-based cancer treatment. So going into some of the data that we have generated. So here we are basically treating a transgenic mice with CITCO, and using 3 different routes. We obtain the liver, and then we we want to measure the expression of that enzyme. Now here you would notice we are calling it a CYP2B10. That is because this is in mouse. So it's basically the human, like mouse version of that enzyme 2B6. So in humans, the enzyme is called CYP2B6. In mice, it's called CYP2B10. So when you look at some of the mRNA data expression, we see induction, like significant induction of that enzyme when we dose the mice with CITCO. And orally, we are seeing it to be as effective as by the IP and the IV route. Then what about the protein expression? Because ultimately that is what is important. So when we do, after our Western blot analysis, we do see like optimum induction of the CYP2B10 even at the protein level. So this is telling us that CITCO in vivo, in mouse, it's working. Next, we want to see the exposure of CITCO in these transgenic mice. And so we obtain the blood samples, we do a bioanalytical extraction technique and analyze the concentrations using LC-MS. And from that, we can see adequate systemic exposure of CITCO, both by the IP route as well as oral. And we see that the concentration reaches maximum at around 4 hours and then begins to decline. So next step is to actually see if CITCO can improve the bioactivation of CPA, because like I said, CITCO can increase the expression of that enzyme, which is responsible for converting CPA to its metabolite. And this is the scheme, how we do the study is we first dose the mice with CITCO, then after 24 hours, we dose the mice with CPA. We collect the blood samples, and now we analyze the blood for CPA and 4-hydroxy-CPA. And the data that we have generated, here you see the serum CPA concentrations. So the mice which receive CITCO, you see, sorry, my pointers. Yeah, so the mice which receive CITCO, you see lower concentrations of CPA in the serum. Whereas when you look at the metabolite, you see higher concentrations of the metabolite in the mice receiving that the inducer, which is CITCO. So this is telling us that the inducer is actually helping improve or increase the bioactivation, the metabolism of CPA to its metabolite. And that's why we get, when you look at the metabolite to parent ratio, we see a higher ratio in the presence of CITCO, further indicating that, yes, this compound is helping enhance the metabolism of CPA. Now, this is all good, but does this mean that the efficacy, which is what really matters, does the efficacy actually increase when you have this inducer compound? And to study that, we use this lymphoma xenograft mouse model, where we actually use mouse lymphoma cells and inject it into these mice, wait for the tumors to grow, and divide the mice now into 4 different groups. The first 2 groups, they don't get any drug. The third group gets CHOP. CHOP is basically a CPA-based regimen which is used in clinics, and the 4th group gets CITCO as well as CHOP. So as you can see, the vehicle group, the tumor volume keeps increasing, whereas the mice which receive only CHOP, you see a decrease as expected in the tumor volume. But what is important here is when you give that inducer compound, the CITCO, you see a further decrease in the tumor volume. That same result is mimicked in when you look at the tumor mass. Finally, when you look at the cell proliferation marker genes, you see that there is a decrease in the expression of these marker cell proliferation genes in those mice which are receiving, again, the combination of CITCO and CHOP, telling us that those cells in those tumors, they are not dividing. And then when you look at the marker for cell death, which is cleaved caspase 3, you see an increased expression in those mice getting that combination drug. So overall, this is telling us that CPA efficacy is improving when you give that inducer. So in summary, what we have shown through our data is CITCO can induce that enzyme, CYP2B10, which is involved in the bioactivation of CPA. So you induce the enzyme, you get greater bioactivation that further enhances the CPA efficacy. And with that, I would like to thank my lab, and especially my advisor and all my lab members, my funding sources. And with that, thank you. And I'll be happy to take questions at the end of the session. Thank you.
Thank you so much. Fascinating presentation, fascinating work. Next, we have Alana Munger and Wei Cheng on multiple myeloma, our first dual presentation, at least on the agenda. There we go. We can see your slides.
Perfect. Thank you so much for having us. And we want to thank the CERSI folks for supporting our research. I can introduce myself. My name is Dr. Alana Munger. I'm a 5th-year orthopedic surgery resident at Yale. I'll be doing my musculoskeletal oncology fellowship at MD Anderson next year, and I'll let Dr. Cheng introduce himself as well.
My name is Wei Cheng. I'm a statistician, and I'm an associate research scientist working in the School of Public Health.
Perfect. And so we are working together to do an updated systematic review and network meta-analysis on bone disease modifying agents in multiple myeloma. And so many of you are probably asking, why is the surgeon talking about medications? Why isn't she talking about surgery? Valid question. So this is one of my cases when I was the chief of the oncology service. And so it was a 69-year-old male who had left thigh pain, but it, basically resolved with a cream that he was using. And you can see this is some x-rays of his femur, and it shows that there's this lytic lesion in the distal aspect of his femoral shaft. And so this was actually a 79-year-old male with newly diagnosed multiple myeloma who had not yet undergone any treatment for his myeloma. So as a surgeon, the questions that I was asking myself was, should I operate? And if so, when? As we know, multiple myeloma, it's a malignancy of terminally differentiated plasma cells. And the plasma cells are these guys all the way at the bottom, where they're basically cells that make a specific antibody. Multiple myeloma is estimated to represent 1% of global cancer burden. And there are 176 new cases each year and 117,000 deaths worldwide. And that was just in 2020. And so multiple myeloma, the good news about multiple myeloma is that our treatments for it have been advancing over the years such that survivorship is improving. And that's thought to be due to the implementation of stem cell transplantation, immunomodulatory drugs, as well as proteasome inhibitors. And during my research, it's crazy how every year it's a new standard for treatment. And so because these patients are living longer, I'm going to be seeing them more. And so this was actually that patient where he actually suffered a pathologic fracture. Multiple myeloma bone disease affects a significant portion of these patients. 60% of them have bone pain at the time of their diagnosis. In a review of over 1,000 patients, 66% had lytic lesions, over a quarter had pathologic fractures, almost a quarter had compression fractures, and many of them had osteoporosis. And so in 2017, the Cochrane Library put out their own network meta-analysis that was performed looking at just bisphosphonates in multiple myeloma. They basically found that it reduced pathologic vertebral fractures, skeletal events, as well as pain. There was an increased risk of developing osteonecrosis of the jaw, but there was not a superiority of any specific bisphosphonate for any outcome. But they did note that zoledronate was sort of edging out the others. Some things to note with regard to that prior study is that it was done in 2017, and the randomized controlled trials for denosumab, which is an antibody basically going after the rank ligand, there have been multiple randomized controlled trials that have compared denosumab to placebo, to zoledronic acid, and that was not included in that 2017 review. That 2017 review also had 4 total databases, but it did not include PubMed, Web of Science, and Scopus, and the network meta-analysis itself was not as robust as our standards today. And so our goal is to perform an updated systematic review and network meta-analysis in order to determine which bone disease modifying agents resulted in better outcomes for patients in multiple myeloma. So we did 7 databases as are listed here. And we had the help of a fantastic clinical research and educational librarian who was able to help gather our search strategy and perform the literature acquisition for us. Our inclusion criteria were randomized controlled trials where it was phase 3 or equivalent. So we excluded phase 1 as well as phase 2 clinical trials. And it had to be a bone disease modifying agent versus either placebo or another bone disease modifying agent in patients with a diagnosis of multiple myeloma. And again, this is multiple myeloma, not the quote unquote asymptomatic multiple myeloma. And the randomized control trial was included as long as it had one of these 4 primary outcomes, which is number of patients with a skeletal related event, time to the skeletal related event, overall survival, as well as progression-free survival. And again, these are the primary outcomes that we have been gathering, and these are the secondary outcomes that we are looking at as well. The exclusion criteria, wrong disease. So we did exclude those with MGUS as well as smoldering multiple myeloma. And this is something that I do think strongly of because for context, smoldering multiple myeloma, 10% of those patients will go to malignant disease, meaning multiple myeloma, in 5 years. So that means that 90% of patients don't get multiple myeloma. They don't get those end stage symptoms. And so the standard of care is that these patients do not get any of the treatment. And so from our standpoint, they're different cells. And so ergo, we wanted to compare apples to apples as much as possible. And so we excluded those patients. We excluded things that had the wrong study design, clinical trials that were not randomized, any incomplete data, duplicate publications. And in talking about inclusivity of studies, we did not exclude any article based on language. So we actually worked with many people to help us translate over 70 articles that were written in almost 10 different languages. And all of those were screened in the full text screening. And we actually have one German article that we're including in the end results. So I think that it was a kind of a kudos to us to make sure that we were as inclusive as possible. This is what we've done thus far. So we had 6,717 references, and 156 were removed because they were duplicates. So we actually did abstract screening for 6,500, and we did a full text review of 920. And right now we have about 71 studies, including in our review. This is a study that's in progress. And so the next work that we need to do is extract the data from our articles, perform the network meta-analysis, and then write and submit a manuscript. Just 3 little bullet points, it's fine. And so I want to transition for Dr. Chang to allow him to speak about the network meta-analysis, which he is an expert in.
Thank you, Dr. Munger. So in case any of the audience is interested in the methodology of network meta-analysis per se, I can give a very brief introduction. So you see a fictional network diagram on the screen, and the nodes represent treatments while the edges represent trials or pairwise comparisons. So in essence, we are, it's not just a meta-analysis for pairwise comparison of 2 treatments. So we may have a collection of many medications for the multiple myeloma condition, and like say we have 4 major outcomes and less than 10 secondary outcomes. So each will have such a network diagram. And for each treatment node, we require at least 2 randomized controlled trials connecting it to the rest of the network. So that could be 2 trials for the same comparison, or it could be like connecting a treatment node to multiple other treatment nodes to form a loop. For example, the red arrow points out to a treatment node that's connected to 2 other nodes. And the purpose of requiring at least 2 randomized controlled trials connecting it to the rest of the network is to avoid a treatment in a single trial connection with a very large effect size. And so this is not like every statistician does this practice. It's I actually will recommend this practice because it's it's very parallel to a meta-analysis in the traditional meta-analysis setting. And we could add in additional analysis or tabulate say such a treatment in a single trial connection in the appendix. Next slide, please.
Mm-hmm.
So I have some reflections on outcomes of network meta-analysis that might be helpful to the audience who are interested in the methodology. So we have most reported outcome is a number of patients with the skeletal related events. That's binomial, which means it has a numerator and denominator. And this is straightforward. And we have the time to event outcomes, and due to the various measures that each time to event outcome could be reported across trials, it might be difficult to perform evidence synthesis. For example, the network may not be well connected for hazard ratio of the time to skeletal-related events. So as a matter of fact, the MASCA 2017 review actually doesn't perform a real network meta-analysis because I think the reason is that their network is not connected enough, well connected to perform a network meta-analysis. And for those 3 time to event outcomes, for example, some trials may have reported hazard ratio for pairwise comparisons with confidence intervals. Some may have reported median or mean survival time to event per arm with confidence intervals and error or standard deviation. Some may have reported a proportion of the SRE-free survival or overall survival or progression-free survival per arm at a certain time point. And the problem is, say, we need to define which time points can be synthesized together as a binomial proportion. So the overlap of, say, not every trial actually reports all these measures for time to event outcomes, which is a challenge for the synthesis. And we may obtain additional trial results as a solution, or say, I would suggest that consortiums or platforms or individual patient level data on each disease or indication specific topic can be established. Say, for example, if say any of the centers want to say establish such a consortium platform for a cumulative or living systematic review and constantly updating it, I would recommend that practice. And I'll hand it over to Dr. Munger.
Thank you, Dr. Chang. And so I think that one of the things that we hope to do is provide a robust systematic review, network meta-analysis that can help to provide evidence to guide clinically based guidelines and from the surgeon standpoint of view, because everything needs to circle back to the surgeon. It'll actually give me time to an understanding of when I would expect a patient to develop that skeletal related event, and maybe that would help me with my indications for surgical intervention. Certainly there are limitations in our work in that we're comparing trials that sometimes are published in different decades. And we are trying to create the best search that we could, but there are always possibilities that there are unpublished investigations. And we can't be sure that all the data of the investigations were actually published, i.e. maybe they didn't want to publish the negative data that they have. And with that, I want to thank everybody, and I will be happy to answer questions at the end.
Thanks so much. Thanks to you both. I should just mention with respect to network meta-analyses, I serve on one of the advisory councils for ICER, the Health Technology Assessment Group, and that is the bread and butter tool that they often use to think about the new products they're evaluating and comparative effectiveness relative to other therapeutic options, given the paucity of head-to-head trials that we know so well. So another great application of the work here presented by this group. Last but not least, thankful to have Mariam Musavi present on her work on zebrafish genetics and anti-senescence therapy. So Mariam, the floor is yours. We can see your slides and we can see you. Go right ahead.
Mariam, you seem to be on mute. We can't hear you, but we can see your lips moving.
Yep. And your thing says unmuted, but but nonetheless, we can't hear you. Oh.
How about now?
Now we can.
Yeah.
Excellent.
Yep. Yes. My project is about develop anti-senescent therapies for anthracycline-induced cardiotoxicity via zebrafish genetics. And first, I'm gonna briefly introduce what is the anthracycline and what is the importance in clinics. So anthracyclines, as all of us know, are among the most potent chemotherapeutics which are used for breast cancer and lymphoma. But its use is restricted because of the cardiotoxicity. This cardiotoxicity induced by anthracycline is one of the worst type of cardiomyopathy, which partially respond to the standard heart failure therapies, including beta blocker and ACE inhibitors. and there's not specific therapy designed to treat AIC. The contribution of the cardiac senescence to the AIC and the signaling pathway remain elusive. So we hypothesize that the therapeutic effect of the autophagy activation in the late phase of the AIC is owing to its antisenescence effect. And there are different types of animal models, such as mice and rats, which are extensively used for the cardiac research, but they're not suitable because of the large-scale screening of pharmaceutical target is very expensive, and due to management colony, it is very hard. So we go to zebrafish, and zebrafish come into play because of its genome, which shows almost 70% homology with human, and it can cover 80% of the disease-related genes. And it can produce approximately 200 fertilized eggs per mating, and their extrauterine growth is rapid, and major organs of the zebrafish are developed very fast by 5 days post-fertilization. They're very tiny, so they require a low-cost diet and also small place, and their optical clarity of the embryo enables us to do the non-invasive fluorescent imaging. And so all of these features enables us to do high throughput genetic variant and modifier screening through F0-based genetic analysis, and the zebrafish can AIC. Since the senescence is one of the 12 hallmarks of aging, we suppose that maybe there is an aging process in the AIC-induced cardiotoxicity. We try to measure some hallmarks of aging in this regard. One of them is cell cycle arrest. The other one is the chromatin regulation, and the secretory phenotype, and the lysosomal compartment. So our first aim is to define accelerated aging process in adult AIC model, and our second aim was to discover new genes, signaling pathway, modify cardiac dysfunction, senescence, or aging in AIC by screening DEG genes using MMEJ-based F0 genetic analysis. The methods that we go through is first the zebrafish husbandry, which is 28.5 degrees centigrade, 14 hours light, 10 hours dark cycle, and use the senescence-associated beta-galactosidase staining, immunofluorescent staining, and quantification for two markers. One is gamma H2AX, and the other one is H3K9ME3. And the third one is quantitative real-time PCR for the cardiac remodeling and the senescence markers, Western blotting for the ERK dynamics, and the last one is transcriptomic analysis to find the genes which can do the F0-based genetic analysis through MMEJ and find the modifier genes in this pathway. So first, we generated a zebrafish AIC model. Since all methods of the IP injection methods are we faced with this and body damage, so we try to introduce a new IP injection method which can be worked by different researchers. And you can see here, researcher number 2, 3, and 4 did the same, and the survival rate was very good. And in order to see if there is a cardiac dysfunction here, we did echo, and by measuring the ejection fraction percentage, you can see it drops, and after 56 dpi of the docs. To validate the aging-related markers, we did the qPCR for cardiac remodeling markers, which you can see the MPPA is statistically significant and higher. And when you compare white lab with docs 56 dpi control, and the senescence markers that we try to find is CDKN1A, which represented P16, and CDKN2A, which represented P15, is actually more than double in this way. And JAK2A and PPARGC2 is also changed and decreased in this way, and as well as the COX-4-1. The other markers for the aging process is to do the beta-galactosidase staining. The beta-galactosidase activity is higher in 56 dpi of the docs injected fish versus hanks. And you can see the gamma H2A positive cells is higher if docs 56 dpi when you compare with the hanks. And this is the marker for the DNA brick, and H3K9ME3, which is the epigenetic marker is also increased in DOX56 DPI versus Hanks's. We also did the quantification for these two markers, and you can see the increased number in groups of DOX56 DPI. The overall conclusion up to this level is that the IP injection of the DOX can generate zebrafish AIC model 56 DPI to reduction of the ejection fraction percentage without incurring fish death and body damage, and increased mRNA level of MPPA as a cardiac remodeling marker, which favors the cardiomyopathy and aging, and increased senescence associated beta-galactosidase staining shows gradually aging in the AIC zebrafish model. And increased mRNA level of P16 and P15 have been accumulated in aged tissue, which favors the aging, and also increased mRNA expression level of COX4I1, which is subunit of cytochrome oxidase C participating in respiratory chain. Therefore, we're facing with reduced oxygen availability, and decreased mRNA level of PPARGC, which examined the mitochondrial genesis and ATP production, can affect this way, and also increased gamma H2AX, which is an indicator of DNA double-strand break, can a marker in cardiomyocyte. This marker is cardiomyocyte slightly increased during cardiac aging, and H3K9ME3, which is an epigenetic marker, is favors for cardiac senescence. And the last one is inflammaging, which we saw increased level of interleukin B in this aging process. This is all the conclusion up to this level, and I want to thank you and Professor Shu and all the lab members here.
Thank you so much. Thanks for all the presentations. What a reminder of the breadth of the kinds of evidence and knowledge and work that can advance the work that FDA does, advance the work that the products that it evaluates can provide to patients and providers. We have a few minutes. We are a little behind, but certainly a few minutes for questions, including questions that speakers may have for each other. That's certainly welcome and encouraged. Joe, I know you need to leave in a few minutes, but I think if you have a quick question, by all means.
Yeah, I mean, I know nothing about basic science, but my question was for Ritika, because I was just so interested in how the sort of augmentation process can work to advance drug efficacy without having any corresponding effect on drug safety. Because I usually think that the sort of the impact would go hand in hand. And so I was just curious sort of how you expect that will work in humans.
Yeah, so we are, like you said, like we are trying to increase the efficacy of CPA by generating more of the mustard. But the problem, like as with CPA, it's a chemotherapeutic agent. And so the mustard basically doesn't distinguish between like healthy cells versus the tumor cells. So that is an inherent disadvantage. But what we are trying to do is if we can activate that enzyme and generate more of the mustard, but without actually increasing the dose of CPA. So maybe even try to lower the dose, then we are getting the same amount of mustard, but with a lower dose of cyclophosphamide. So, and also in some cases, some patients don't have enough of that enzyme, which can result in that generation of that mustard. So that's the whole reason why we are trying to like basically look into this. Thanks.
Other questions for any of our presenters? I know it's been a full afternoon of lots of good knowledge to think about. Seeing.
I actually have a question.
Please.
Sorry, it's for Mariam, and it's very interesting because we also work on dogs related like cardiac toxicity. So I wanted to know, did you all look at the reactive oxygen species levels? Because that's the marker that we use typically. So I just was curious about that.
No, not yet. We didn't have this chance, but this is very good to just look at this also marker as well, because it can also affect the aging process, I think.
Thanks.
Excellent. Any other questions on folks' minds?
No, I have to run on. I just want to congratulate all of the scholars on amazing presentations, great work. Some of it in process, some of it, it's really just terrific to see the breadth of work that's happening across all the CERSIs by so many different folks. And one thing that we will do is we'll package your slides as PDFs to send to folks at FDA. Not everyone can always attend these things, and there often is a interest in hearing more. So as long as it's okay with everyone, we'll confirm and check. But Molly, I don't know if you have any closing comments, but I hope everyone has a great weekend, and thank you again for making time to present today.
Thanks, Joe. Yeah, nothing super groundbreaking, but I do just want to thank you all as well for joining us today. Really, as Jason said, one of my favorite things about this showcase every year is really just the breadth of expertise and the breadth of methods that people use, from bench science, statistical methods, evidence synthesis, data science, artificial intelligence, and I mean, literally everything in between. And so we really appreciate you and congratulate all of you on the work that you're doing. One thing I would also like to say is, one of the greatest pleasures of doing this work is that we get to work directly with our primary audience, our colleagues at the FDA. And we just appreciate so much that you joined today, or that you're watching this webinar after the fact. I'm really excited about some of the connections that we saw made between the scholars and folks at the FDA. And please do reach out. We will, as Joe said, be sending out the presentation so you can reach out to people. But thank you to our colleagues at ORSI, thanks, and across the FDA for all of your partnership in this work of the CERSI. Thank you to all of the scholars and mentors who participated this year. And I also, last but not least, want to thank Michelle Bernabeo for her really outstanding work pulling this day together. So thank you all for coming, and we will see you next year.